Okay, good morning. Welcome back to the Delta Independent Science Board's Food Web Workshop, second day. I hope you all had a good evening and maybe saw the Kings win their game 2 blocks from here. Uh, so I'd like to call the meeting to order, and we'll do the roll call next. Are you ready, everybody? Could I ask the ISB members to stand up and just go like we did it yesterday, just go through the room.
Steve Brandt, no new declarations.
Kenny Rose, no new declarations.
Bob Nyman, no new declarations.
Diane McKnight, no new declarations.
Tom Holzer, no new declarations.
All right, so we'll go to the members that are online. Tanya, can you take over?
Anya Heikola, no new declarations.
Anna
Anna Mikulak, no new declarations.
and Obi,
Jayant Abheshekar, no new declaration.
Virginia,
Virginia Dale, no new declarations.
and Lisa. I'm sorry you're still sick.
Thank you for handling everything, Inga. Lisa Wanger, no new declarations.
And Inga Vernon, no new declarations. And with that, I hand over to Steve Brandt. Oh, public comments. Do we have public comments?
There are none.
Welcome back, everyone, or welcome to those who couldn't make it yesterday. I'm not going to remind you much about what the overall purpose of our workshop was, but for those who couldn't make it yesterday, or those who really want to suffer, I say that jokingly, these meetings are being recorded and they're available on CalSpan. So at any time in the future, you can go back and listen to everything we've said. So I think that that's a good chance to review, particularly for those that couldn't be here yesterday. So I've been given the charge of putting a recap together of everything we discussed yesterday. And our primary discussion yesterday was focused on trying to look at how food web understanding, what sort of management issues are there where food web understanding could help either inform those management decisions or help predict what the consequences of changes in drivers or management decisions might be to the ecosystem. And as I think you all know, we had a number of talks looking at other ecosystems, looking at panels, looking at their perspective on what some of the fisheries management issues might be, when some of the ecosystem management issues might be, and then breakout sessions where we could all have a chance to say, okay, let's dive into it a bit and look at what might be happening. Now, those discussions, in my view, were extremely rich. There was tons of ideas, and I was charged today to recap all of the conclusions. Bob was supposed to give it last night, but he gave it to me to do it this morning. So I gave it to Lillian to put together one slide that encapsulated it all. So this is largely from Lillian, and but I think she handled it, did it very well. I think we discussed a number of issues that require a better understanding of food webs, and I think we have a list of those and why they might be important. And I think that even one person said everything that's happening in the Delta requires looking at the food webs. And so I think I'm more leaning in that sort of direction as well. All of this stuff will be summarized. We have sheets of information on that. And when we write the final report, we'll try and encapsulate all of the rich ideas that came out of all of those various presentations and discussions. Some of the other things that came out is that the concept that many of the I guess, the regulations or focus in that is on a few species that happen to be threatened in the system. And I think it came out a lot of places that those individual species are so rare that they probably don't have a huge impact on the food web per se, whether it's biomass or energy flow in that because of their rarity. But in contrast, the other species that make up the biomass probably have a significant impact on the food of these rare species and or on their predators in terms of providing alternate food that could either help those predators do well and so forth. So that's sort of the connection there. There was a suggestion that or two that exploring ensemble modeling approaches that are broad might be one approach to way of moving forward. Like there's no single answer, but there might be an ensemble approach in terms of modeling. That there was a suggestion to create a vision of a preferred future delta rather than the delta of the yesterday by applying forecasting and scenarios, maybe to evaluate the impact of future management actions on food webs. Clearly, there's been a lot of visionary work done in the Delta. So some of that stuff is well established and so forth. Maybe having a look at that in terms of food webs per se might be a valuable approach. And then there were some suggestions that all of this, any future action or future research or how we move forward on this is clearly going to require some sort of thinking about how it's funded. And so that's some of the ideas. And I think anyone else who was here would probably come up with a completely different list. So I think that this is just some ideas that came out of it. Yesterday, we talked about the relationships to management. Today, the first part of today, we're going to really talk about looking at the individual tools and approaches. So we'll have a series of presentations and lightning talks that kind of dive a little bit deeper into some of the approaches of doing food web analysis or food web studies or better understanding food webs. And that will we'll actually have then a breakout session following that will ask specifically the question, now, given that we know what some of the management issues might be and what the array of tools might be, what might be the most valuable tool that would have applicability in the Delta? And try and come up with a list of some of those tools that might be most appropriate or most effective being used in the Delta. And then we'll end the day by looking forward. We'll have Peter Goodwin give a inspirational talk on looking forward to the future and how we might be able to move forward on this. And then the breakout groups will be very specific. In this case, we're going to ask the breakout groups to come up with sort of a prioritized list of the 3 next things we need to do in order to move forward on the science of this topic within the Delta. So with that, I think, again, to remind folks that to think big and be creative and be optimistic, think strategically, and looking forward, it might be possible. And so with that, I think we're going to move into the first presentation. And the first presentation is by Ryan Bellmore. And I will also remind folks I'm going to do what Bob did yesterday, as I will stand up when you have 1 minute left to talk, and then you'll be eating into your question time for the speaker. So, Ryan.
So I'm assuming like I have to stand in front of this. Okay, just like I'm gonna wander, so just make sure I come back here. I've never been on CalSpan before and had to stay in front of the mic before. You guys hear me okay? Yeah. Wonderful. And does this work?
It's a pointer, but
It won't advance slides.
it won't
Okay.
advance slide, yes.
It will?
Yeah.
I have to point it at the. Which one do I push?
Technical demo.
Oh, there we go. Oh, that's great. Okay. Well, thank you guys for inviting me here to speak today. It's I was saying last night that I'm almost infinitely more knowledgeable about the Delta than I was 48 hours ago, which I basically knew 0 about the Delta. And it's also about 40 degrees and raining in Juneau right now. So this weather is just wonderful to get a little bit of sun. So today I'm going to talk about that is my perspectives, I should say, and also some approaches for studying and modeling aquatic food webs, or what we sometimes refer to as the invisible fabric of nature. And we study food webs for a lot of reasons. We study food webs to understand patterns of productivity in ecosystems. We study food webs to help identify features of ecosystems that promote ecological stability and the maintenance of biodiversity. And of course, we talked about this yesterday. We study food webs to help us understand how to manage whole ecosystems for the parts of those systems, the species that we care about. That said, food webs can seem at times almost infinitely complex. So given this complexity, how do we study food webs in a way that we can use to inform ecosystem management? And I think there's three things that we need to include when we do this. One is we need to have empirical studies. And I think sometimes we need to have pretty detailed empirical studies of the real food webs that we're interested in. We also need models. And I think these models, at least some of them, should be pretty tractable and comprehensible that we can use to simulate how food webs might change with environmental conditions or management actions. And importantly, we need some process to connect these two, right? So whereby we're using our empirical studies to inform the structure and assumptions of models. And we're using our models to generate hypotheses, to guide our observations, to guide our experiments, creating this adaptive modeling feedback. I highly recommend this paper by Mary Power, who talks about this process called Field Biology, Food Web Models and Management. Happy to share that with the group if anyone's interested. So today, I'm going to talk about, again, some approaches and perspectives for one, empirically examining food webs, constructing empirical models of food webs, two, modeling, creating simulation models of food webs to predict how food webs might change with environmental change or management actions, and then some coming back with some high-level perspectives on sort of linking models and empirical observations together. So we talked about this a lot yesterday too. When you study food webs, you're confronted with questions about complexity and scale. How much complexity of the rich species and interactions in those food webs do we represent, either in our empirical studies or our models, and at what spatial and at what temporal scales do we represent them? So in terms of food web complexity, for instance, do we aggregate all the species into a simple food chain, or we disaggregate those species to represent that rich diversity of organisms and interactions? Or do we in terms of spatial scale, do we study food webs at a single location at small scales, or do we study food webs at broad spatial scales, representing the heterogeneity in the landscape that's important to many species? In terms of temporal scale, are we aggregating or averaging the dynamics of food webs and environmental conditions that affect them over long time scales, such as years? Or are we trying to elucidate food webs at finer, seasonal, monthly, daily scales? Of course, the answer to these questions depends on the research questions that we're asking. But I do think that sometimes there's an assumption that we want to be over here. We want to be over here as far as we can get into the complexity side of these. And I think, one, that can be really difficult to do in the context of one or two empirical studies. And from a modeling perspective, it may not necessarily be the best thing to do to be over here as far as possible on these complexity scales. And I'll talk about that a little bit more in a bit. So now I just first want to talk about, which will be a lot of the talk actually today, it's more of my background, some empirical approaches to elucidating food webs, sort of using these different gradients to guide our conversation. So when we're building a food web empirically, there's two axes of food web complexity we need to consider. One is this axis of taxonomic resolution, right? Like how much of the rich diversity of species are going to represent in the webs. Are we going to aggregate the food webs into trophic levels, right? Food chain. Are we going to aggregate it into different functional groups of organisms? Or are we going to break it out and really represent the diversity of different species that are in that web and associated interactions? Again, it depends on the question we're asking. In many cases, in fisheries studies, what we're doing is we're aggregating organisms down here at the bottom of the food web, like phytoplankton, zooplankton, detritus. We're lumping those together, and then we're breaking out the higher trophic levels into functional groups or the specific species that we're interested in. And then the other axis that we need to think about is how much we're actually going to quantify those linkages between the nodes in the food web. So down here, the simplest way to do this is to create just linkage webs, right? That those links are all weighted the same. They're 0 or 1. That just depicts whether a feeding link is present or absent. But we can step up in complexity and we can start weighting those linkages. So the next step up would be to create like diet composition webs. In this case, the linkages are weighted by the contribution of different prey items to the diet of those different predators. And then the next step up in complexity would be constructing, I think we talked about these a little bit yesterday too, flow webs that represent the linkages are weighted by the amount of organic matter or energy flowing along a given pathway. So each one of these linkages represents how much food that the different prey items that a consumer is eating in those webs. So you can sort of think of these as, what are you eating? What are your favorite foods? And how much of those foods are you eating? So I'm going to go through and talk about some of the advantages and disadvantages of these different approaches and some very high level thoughts about how you construct these different food webs, starting with these linkage-based webs. So the advantage of developing a linkage-based web is that, one, they are, in some ways, they can be relatively easy to construct. And two, there's a whole series of papers applying network analyses to these linkage-based webs to understand the connectance of these food webs. There's tons of metrics you can calculate that tells you about the connectance among species. And they can say something about the potential stability of these webs. And if you can do that, then you can examine how changes to the structure of that network. So for example, removing a species from that network could ripple throughout the system and affect those other different connectance metrics and the potential stability. But again, the disadvantage of these webs is that all the linkages, they're weighted exactly the same. You can't tell which linkage is more important than another linkage to any given organism. Like I said, they can be relatively easy to construct, but also many of these linkage webs, as you can see from the previous image, are quite complex. Usually they're constructed using diet analysis for different consumers in the web, and doing that in the traditional sense of IDing these organisms in the lab, or I think these linkage webs are this newer DNA metabarcoding and such approaches are really well suited to this. And isotopes can also be used, especially for lower trophic levels, to sort of piece these things together. So the next step up in complexity is these diet composition webs. And in this case, again, we're beginning to weight the linkages in the food web now. And so we're weighting the linkages by the importance of each resource to the diet of each consumer. And thus we can start ranking the importance of different prey items to individual consumers in the food web. And these can be pretty complex, obviously, like here. I think this was probably constructed with diet information, isotopes, or they can be pretty simple as this lake food web that's constructed with isotopic information. But there's also some disadvantages to these webs is that you can't actually tell how much of these different food resources consumers in the web are actually eating over a given period of time. And that type of information really helps you understand the potential for food limitation, top-down control, competition, et cetera. Okay, so these diet composition webs are generally constructed by calculating the contribution by biomass of different prey items in the diets of consumers. And again, we often do this using just like traditional approaches where we're looking at what's in consumer diets and calculating these proportions. But this can also be done with isotope analysis. And there's pluses and minuses to these different approaches, right? If you diet analyses, if you like taking stomachs out of fish, for instance, give you really high taxonomic resolution of things that are being consumed, but it's only a snapshot in time. Whereas isotopes are integrating diets over much longer time intervals, depending on what tissue you're using in those analyses. But they're generally low in taxonomic resolution that you can identify. It's more about what are the energy channels fueling, consumer production. So you can see there's pluses and minuses to each approach, and there's actually there are a lot of strength to be gained by combining diets and isotopes into single studies to do this. Another benefit of isotopes that list up here is that they're not telling you just what they ate, they're telling you what they assimilated. So it's telling you something about the quality of prey. Okay, flow food webs. The advantages of this approach is that, well, I like it, and I've done a lot of it, but in this case, the links are weighted by the amount of energy or carbon flowing through the food web, or even potential contaminants. Talk about that in a second. So again, in this food web, you can see that the links are weighted by organic matter flow, grams of dry mass that are moving between these insects down here to the fish up there on a per meter squared unit of stream bed. This is from a stream over a year-long period. And if you can do this, well, then you can start mapping these consumptive pathways that support the species that you're interested in, and also mapping the consumptive pathways of all the other species that are interacting with those species you're interested in. And you can use this approach to calculate a variety of management relevant metrics, such as food limitation, competition for shared food resources, and sort of food-based carrying capacity estimates as well. I think one of the other advantages of this approach is that you can begin to identify food web features that are theorized to promote the stability of food webs. We talk a lot about productivity of food webs all the time. We have sort of a productivity bias, but stability is really important. And a couple of things that are theorized to promote stability is the prevalence of multi-channel feeding. So if for instance, consumers that are coupling detrital energy channels and algal energy channels, as well as webs that have a lot of weaker interactions. Again, we focus a lot on these strong interactions, but there's been a lot of studies that have shown these weak interactions are actually what stabilizes the web, because these strong interactions can be stabilizing, so they sort of counterbalance those strong interactions. This is a food web, a flow food web we constructed for a stream, a salmon-bearing stream up in Alaska, where I live in Southeast Alaska. And you can see both of these mechanisms where we have the marine-derived nutrients from salmon carcasses, periphyton, vegetation, and detritus, all contributing to the production of these insect consumers here in the middle. And we also see a lot of these weak linkages through the food web again, and both of these mechanisms are thought to be stabilizing. The downside of this approach is that it can be quite difficult to construct these. You need a lot of information, and in some cases you have to have a lot of assumptions to put these together too. You need production estimates for your different consumers in your web. And I say production estimates, I mean how much biomass are those different consumers accruing over a given period of time. A lot of times we build these over a year, so well, how much biomass is a given fish producing over a year? And we need to combine that with consumer diet information. That can be either from diets, lavage, stomach contents, or isotopes. And then you combine that with bioenergetic modeling, so you can actually tell how much of these different prey resources fish are eating to fuel the observed amount of production that you estimated. And by the way, Dave Beauchamp is going to talk about bioenergetic approaches, I think, in just a few minutes. In freshwater ecosystems, these flow food webs have frequently been constructed using an approach called the trophic basis of production approach that was originated by Arthur Banky and several of his colleagues, and has been used a lot in freshwater ecosystems around the world. I just really quick want to tell you about one of my favorite examples of this approach. Was from the Colorado River, where they used a flow food web approach to understand how high flows. They did these high flow experiments in the Colorado River. There's a lot of news stories about that. And so they used the flow food web approach to look at how those high flows change the pathways and magnitudes of energy flow, supporting insects and ultimately rainbow trout. And they found this somewhat paradoxical response at first, which was that trout production doubled after the flood, yet the production of their primary aquatic invertebrate prey in total went down by 60%. It's like, well, how did trout production double then? And what happened was that there's these two chironomids, so black flies and midges, that didn't represent that much of the total insect production, but they were really important to fueling fish production. And after the high flow experiment, the production and consumption of those two pathways in the food web increased by 300%. So despite the fact that this whole invertebrate assemblage, the production decreased by 60%, these two organisms substantially increased in their production and availability, which fueled that doubling of trout production following the high flow experiment. And it would be really hard to have teased this apart without an approach like this. And in turn, this actually increased the fluxes of mercury through the food web to, actually, these are non-native rainbow trout. And so I just wanted to show this really quick, because these flow food webs can be used to trace contaminant flow through food webs if you know the concentration of contaminants in the different consumers and resources in the food web. And that's what they did in this paper. Okay, so let's talk a little bit about what spatial and temporal scales do we think about elucidating these food webs. So the historical approach generally was like we measured food webs at sort of one location in space and sort of one point in time, right? And as we've gained a better appreciation of sort of in landscape ecology, we realized that most landscapes are very temporally heterogeneous and very dynamic in time, and that this can create this patchiness where we have a diversity of different food webs that vary in their structure, vary in the pathways and magnitudes of energy flow that support consumers, and they also vary through time. They exhibit different temporal dynamics, and thus, in many cases, our food web sampling does indeed need to be spatially and temporally explicit to some extent. But the question is, well, what scale do we incorporate that spatial and temporal heterogeneity? Well, in aquatic ecosystems in general, we see this pattern, right? Where the higher trophic levels generally operate over longer time and broader spatial scales than lower trophic levels. So a diatom, freshwater diatom, for instance, may have a turnover time of just a few days, and the space it occupies, if it's a detached diatom, may be only a few millimeters. Whereas organisms higher in the food web, like fishes, live years to decades and utilize a whole diversity of different patches and habitats to complete their life cycle across, and sometimes quite broad landscapes. So I think a good rule of thumb is that the spatial and temporal extent and resolution of our sampling should match this, at least match the scales relevant to the species that we're interested, species or trophic levels that we're interested. And then I think we also need to consider to some extent the spatial and temporal scales relevant to the primary prey resources for those species that we're interested in as well. So one example of this, which thank you, Stan, for highlighting this yesterday, was some work we did up in the Methow River in northwestern Washington, where we were quantifying the food webs and energy flows that supported endangered Chinook salmon and steelhead across this floodplain mosaic, across this whole mosaic of different habitats. And so to do this, we sampled this mosaic of habitats, main channel, all these different off-channel habitats, and we sampled these habitats at monthly time intervals. So it's a time interval that's relevant to both the consumers, the fish, and a time interval that's relevant to calculate the production of their primary prey, which was aquatic insects. And by doing this, we were able to construct these flow food webs that showed the flows of energy and carbon through the web over a year-long period that supported salmon. What we found was there's this like diverse energetic landscape, different habitats supported fish in different ways, this floodplain food web mosaic, if you will. But we also need to recognize, of course, and we know this, that these food webs are not isolated from one another. They're connected via the movement of predators, prey, and resources, creating larger and more spatially structured meta food webs, if you will, or essentially webs of webs. And this has been conceptualized recently as a foodscape, at least for mobile fish consumers, that landscapes or watersheds in this case, and this is a paper we've been working on that's being led by Gabe Rossi at Berkeley. Carson Jeffries is also participating in this, but we've conceptualized this idea of a foodscape that from in a watershed, from headwaters all the way to the nearshore marine, these watersheds support spatio-temporal variability in the abundance of food, in the accessibility of those food resources, and in the energetic costs of accessing those resources. And thus, a landscape can have these diverse foraging and growth opportunities for mobile consumers that can move between these locations on the landscape, potentially tracking asynchronies in foraging and growth opportunities. And I want to acknowledge that actually a lot of the thinking behind these foodscape ideas is from this region. In fact, from the Delta, or maybe it's categorized as just upstream of the Delta, with folks looking at a variability in food webs between main stem and different sort of off-channel habitats and how they support salmon production. So if we're going to think about these meta food webs or foodscapes, well, we need to know something about how sort of consumers and even resources potentially are linking across these different patches, right? And we do have some approaches to do this. I just want to highlight really quick. There's sort of classic sort of tagging approaches, right? We can put pit tags or actually, radio telemetry tags and sort of track how fish are moving. We also just do this observationally. If we see that there's a lot of fish here and then they disappear, we can sort of piece those movement patterns together. But we can also do this implicitly by looking at the isotopes of different body parts. There's been some great research in this region of the world using eye lenses to reconstruct diets, and I think this could also be used to reconstruct sort of habitat occupancy. And of course, we can also look at the microchemistry of fish ear bones or otoliths to reconstruct where they're growing at different times during their life history. So we need to account for food web complexity and spatiotemporal variation relevant to the research questions and species that we're interested in. And again, sorry, this is like a confusing, I don't know why I put this, it's like a 3D plot of like food web complexity goes up here, temporal resolution goes up here, and spatial complexity goes up this way, right? And so we may want to be sort of in this quadrant up here where we're calculating all of these things in very high levels of complexity and resolution. And maybe we do need to be up there, but we can't do that all at once, right? We have to think about creating this portfolio of different studies to be able to recreate that complexity, where maybe in some locations we're constructing these very detailed food webs, maybe using a flow food web approach, but that's really difficult to do. You can't do it everywhere. So maybe you pick a few locations where you develop these, and then we use your knowledge from that to determine how can we simplify this web to understand its sort of key ingredients, and then do that type of sampling at broad spatial scales. And then maybe at other locations, you're doing really fine detailed sampling to understand the temporal resolution of those food webs. And you can piece these different components, this portfolio of studies together to understand the complexity of these food webs in space and time. Okay, so I'm going to switch over to, how much more time do I have? Really? You're not standing up yet. Okay, I'm only at half? Oh, a minute and a half. Okay, well, I don't have very long. So some very quick perspectives, but everyone else went over yesterday. So on modeling food web dynamics. And so again, talking about mathematical food web simulation models, where we're representing usually these food webs as a series of linked sort of differential or mass balance equations. And a lot of times we're linking the rates in those mass balance equations to environmental conditions like water temperature affects physiology and of organisms and production and things like that, and flow and how water is moving and nutrient availability and many other things. And then we're using those models to sort of examine dynamic responses to different model experiments, right? That represent ways we might want to manage the system. And again, just like empirical sort of food web models, there's this gradient of complexity, right? You can represent these in simpler or complex ways. So at one end of the spectrum, we might have like sort of food web archetypes, like represented as simple food chain models or things like that. And then this side, we might have these very complex networks, right? That you're trying to sort of represent all the species in the web and high levels of resolution. You're doing that across space and time and sort of linking that model to tons of different environmental conditions that influence the web. And as you can imagine, these different approaches have different strengths and weaknesses. This end of the spectrum, these are really heuristic models, right? They're good at facilitating understanding. They have clear assumptions, but they don't necessarily represent site-specific conditions. In many cases, they're inadequate to inform management decisions. Might be a good place to start just to think about the system, but that's about it. These complex models, on the other hand, they do account in many cases for a lot of site-specific context that you can use to simulate very detailed management alternatives, but they can sort of suffer from the sort of black box syndrome, where it can be really hard to interpret the behavior of the model and why they're producing the results they are. And in many cases, they have numerous assumptions that can be unclear to the people that aren't building those models. So where do you start, right? So you want to build a model. I think there's often, again, assumption that Like we want to start at the really complex end of the spectrum. It's like, we're going to build one big model and it's going to represent everything, this one model to rule them all sort of paradigm. My philosophy is you should start maybe on the simpler side of the scale. So build the maybe oversimplified in some ways. Build the simplest model you think you can that can represent the key elements of the system to address the questions you're interested in. This is a great quote from that Mary Power paper I said earlier, highlighted earlier. Simple models are testable, discardable. They're not replete with invested effort, so they're easy to reject when they're falsified, and more transparent to scientists and non-scientists alike. So if you start by building a really complex model, what Mary is saying is that you've invested a lot of effort in that. And if it doesn't work, because all models are going to be wrong, that you're going to continue to double down and try to use that model despite the fact that maybe you should reject that model and move on to something different. So I'm not going to go into detail on this because Stan talked about it yesterday and I'm out of time, but I think one example of this is the aquatic trophic productivity model that Stan talked about yesterday. Was very generalized in structure, just represents sort of the generalized trophic structure of river food webs in the Pacific Northwest and many other river food webs in this part of the world too. And we've used this model for a variety of contexts. It's been really useful for understanding responses to dam removal, different river restoration strategies, even climate change. Also, we've used it to understand how nutrient supplementation via carcass addition could affect pathways of energy incorporation of the food web to affect endangered salmon and steelhead recovery. But again, specific predictions of these models are going to fail, right? It's part of the modeling process because there's going to be inadequate complexity in the model, incorrect assumptions, but they're useful because we can learn from, by studying why those predictions fail, we learn something, right? So we're going back to our sort of connections between empirical studies and models. We're using these models to generate hypotheses. We make observations and we use those observations to inform how we change the structure and assumptions, or maybe just start over with an entirely new model or develop a portfolio of models. So you're creating this adaptive modeling feedback. And if you do this, then you can start making sort of targeted increases in the complexity of your model as we increase our sort of understanding of the system. So again, we need to develop studies at appropriate levels of complexity and scale. And I think my philosophy anyways, partying on you guys, is that I really like the idea of like having empirical studies, some of them that are highly detailed to really understand the complexity of the food webs that we have now, and then coupling that with at least initially pretty simple models, and then connecting these two through this adaptive framework. And by the way, you need a lot of different people with expertise to do this. You need silly field scientists. You need modelers. You need people that can do both things, that can go back and forth, right? And of course, you need the buy-in of the managers and practitioners that you're going to want to use this model. So I'm way over time probably, so I'll stop there and have no time for. I'll leave this quote up, which is one of my favorites while we're moving on. But thank you.
We do have time for a question or two. Any questions?
Hi, Rosemary Hartman, Department of Water Resources. Excellent talk. Really enjoyed it. Really like what you're saying about the difference in complexity, and just thinking about, the higher end of complexity, which clearly you've done some of these really complex models. At what point, in my experience, you need so much data to go into those really complex models that it can be easy to build a model with a lot of complexity that doesn't, if you don't have enough data, you don't capture the full range of stochasticities. So you don't necessarily, I guess, what am I trying to ask? Where do you find that the more complex models can break down? What are the potential weak points as you increase complexity, and how do you conquer that?
Right. I think one of the things brought up too is that like the complex model not only the complex models can not only be difficult to understand. I think that's one of the limitations for me is I've not actually done a lot of complex models. The one that I have done, it's like, I can't understand why it's producing that pattern. And thus, and they can be wrong for, it's Dan and I were talking about this, just a complex model can be wrong for potentially hundreds or thousands of reasons, whereas a simple model could be wrong for a much fewer amount of reasons. So you can help understand why your model is wrong, which can help you falsify the model and improve it. And you bring up another aspect of that is that they can be harder to parameterize, right? That you're representing all this complexity. And in that case, you have to make a lot more assumptions about different process rates or turnover times for different organisms in your web or how daily variation affects them. So that's another way that you can sort of get in trouble with that is it takes a lot of data to do it.
Yeah, Ryan, you also mentioned too, which I think is really important, that the food web model you target needs to be in line with the question you're trying to address. And I think that can help determine what the complexity is, because I can see in some of the areas where you were showing these strong interactions, if you did a simple model but forgot one of the major strong interactions, then the model could be really wrong. So the simple model assumes you really know what the fundamental where the fundamental lines are.
Yeah, it does. And I'm not saying like we shouldn't have complex models. I'm just saying it might be better if you're starting from scratch, and you want a model, again, start with the simplest model that you think can represent, the key processes of the system relevant to the question you're asking, and then build in complexity.
Okay, we have one online question from Virginia Dale.
Oh, hi. That was a fabulous talk. You covered so much. But I wondered about the role of hierarchy theory and what you're talking about with scale. Hierarchy theory says you need to look one scale above and one below that of interest to understand the processes of interest. Have you thought about how that may relate to the models you're talking about?
Yeah, that's a good question. I sort of getting at that with sort of thinking about the temporal and spatial scale, sort of sample one. I'm looking down at the screen at you, Virginia, but looking sort of one level in the hierarchy lower than the primary prey rhesus you're interested in. But that's an interesting thought. That's like, different processes are operating at different scales. And do you represent, you can also get in trouble by trying to represent in a single model a lot of different processes that might be happening at scales that are orders of magnitude different from one another. So you're trying to couple processes that are happening on monthly timescales with processes that are happening over hours and seconds in the same model. It can get pretty tricky. You can do that, obviously. So in some cases, at least initially, maybe it makes sense to sort of break those levels of the hierarchy out and use the outputs of one model to sort of feed the other one instead of trying to incorporate all those levels in the hierarchy into one model right away. That's my initial thought on that, Virginia. But good question.
Well, thanks, Ryan. I know there's more questions, but we're out of time. So great talk. Move on to our next speaker, Zachary Emerson.
Good morning, everyone, and hello again. I'm grateful to be with you for a second day, and I'm going to be talking a little bit about food webs in the Delta from the indigenous perspective. My name is Zachary Emerson. I'm the Ecological Heritage Specialist for United Auburn Indian Community, which is a local tribe consisting of Nisenan and Miwok background. Our ancestral territories span quite a large area, including Sacramento, American River, all the way up to Tahoe, all the way to El Dorado County, and all the way to Yuba County. So the Nisenan and Miwok have a deep ancestral relationship to this land, and I'll be sharing with you a little bit today our perspective on some of the management priorities centering around Indigenous knowledge. So a little bit about my background. I'm a restoration ecologist by training, but my passion is really for the synthesis of modern restoration ecology and traditional ecological knowledge. Really, fire is at the core of Indigenous ecosystem management. And for that reason, fire is also a very big focus of my work. I wouldn't be here today if it wasn't for my teachers, elders like Diana Almendirez, Pam Gonzalez, Ardith Reed, and my counterpart at United Auburn Indian Community, our Tribal Historic Preservation Officer, Matt Moore, who co-founded the Coyote Crew Ecocultural Revitalization Program with myself. So I'll just be talking a little bit today about what ecocultural revitalization is. I know that might be a new term for some folks in the room. And really, it just exemplifies that connection between culture and ecology that is absolutely integral and intertwined with everything that we do and the way that we understand our environment. So, as I said, the Nisenan and Miwok people and their cultures are still here, but like native deer grass, for example, we cannot flourish without fire. And deer grass is just one example of a native fire adapted plant. I'll be touching a little bit more later on in the presentation about the specificity of benefits that fire provides, especially in relation to these wetland plant communities that are essential to the Delta. So again, ecology is culture, culture is ecology for Indigenous peoples, and therefore the health of Indigenous peoples is directly dependent on the health of ancestral lands. So when we talk about ecocultural revitalization, it's a very dynamic, all-encompassing term. It's the restoration of relationships to ancestors, plant and animal relatives, and Mother Earth. So this is a little bit about the Coyote Crew. I'm gonna really skim over this because I'd like to spend most of this talk talking a little bit about the specificity around management in the Delta. But this is really our way of building capacity for facilitating the paradigm shift and incorporating more Indigenous management into large-scale agency projects that have been really devoid of tribal partnership in the past. So our Coyote Crew is named after the Nisenan myth of coyotes stealing fire and bringing it to the people. And so we like to joke that we're stealing fire back from the agencies for the sake of all relations. Again, a little background, the crew is relatively recent in its formation, but we are really exemplifying this approach of finding a balance between the new and the old ways. You can see all of our tribal members are wearing full Nomex, protective gear, but we are doing the work with the right intentions and using the traditional knowledge that has been passed down for many, many generations. So, we do use modern equipment and modern methodology because unfortunately we're living in a novel ecosystem now. We've seen, hundreds of years of fire suppression, and the fuel loading in much of our Sierra is now at the point where it makes it very prohibitive to reintroduce cultural fire in purely traditional methods, especially at the scale that we need to reintroduce fire. And one of the things I'd really like to mention during this presentation is the connection between fire and water, because it's something that we often miss. And, there's so much conversation over water diversions within the Delta. But the interesting thing is that we don't examine the biological pathways upstream of the Delta that are contributing greatly to the hydrology of the rest of the watershed. So the picture you actually see up here, we're doing meadow restoration, and we came through and did a considerable amount of conifer removal. This is a Sierra and wet meadow that has experienced a significant amount of conifer encroachment and invasive species encroachment. And so what we've seen is that the increase in conifer dominance is actually greatly depleting the water resources in many of these areas. And when you amplify that change in fuel loading and conifer dominance across the landscape, across the whole Sierra, the water costs of this lack of stewardship of our forests is incredible and can't be overstated. Again, a little bit more about our vision. We're really focused on tending to cultural sites and revitalizing at these places of such significant importance. But really, as I mentioned, the vision is to bring indigenous guardianship back to the ecosystem scale. As I briefly touched on yesterday, historically, pre-contact cultural burning was at the watershed scale. You were using ridge tops, changes in plant communities, topography, geology, to actually develop your burn units and your control lines. And so that's really the scale that we need to get back to. And it has to start with these cultural sites, because these are some of the remnants of culture that have not been destroyed through genocide and development. And so, we have to do a lot of preservation. And unfortunately, we're so caught up trying to preserve the last remnants of our culture from being destroyed on this landscape that we often don't get to focus on restoration. And so I think many tribes are entering into a new era where, through our tribal monitoring programs, we've been able to develop a foundation of preservation so that now we can actually begin to revitalize and restore. Again, I will just speed through this, but we have some great collaborators at UC Berkeley, specifically Professor June Sinceri. And another example of how we're integrating the new and the old ways. We're, as I mentioned, tending to these sites, identifying and understanding them, reclaiming and cleansing them. And then the process of tending and restoration can begin so that we can begin to reconnect the sites to the greater landscape. And obviously, passive restoration needs to be undertaken before active restoration so that we can understand what's still present on these sites, what's still in the seed bank. And that's why fire plays such an important role. It can help to lay the foundation for the active restoration process. It's, far too often people jump the gun and start purchasing seeds and using copious amounts of herbicide, unfortunately, in restoration, when really these sites are just crying for the reintroduction of fire. And we have dormant native seeds in the seed bank that are waiting to express themselves given the proper conditions. Don Hankins at CSU Chico has some amazing research on star thistle. And he was actually able to convert an area that was almost 100% cover star thistle to 100% native cover with no herbicide, no reintroduction of propagules, simply the reintroduction of fire and strategic annual timing. You don't see a major drop in star thistle populations until year 3 to 5, which is why consistent long-term management is so important. That's what so many agencies get wrong. They pat themselves on the back when they do one burn every 10 years. And sometimes that can actually do more harm than good. So here we're just using ground penetrating radar. We also use LiDAR photogrammetry to elucidate these sites. And the cultural fire is very important because GPR does not function very well when you have 6 inches of duff. So the cultural fire actually helps to not just begin the restoration process, but it also expedites the research process. So speed through this as well. What we miss in a lot of these food webs is the human element. And we're so focused on, the health of our animal and plant relatives that we forget that their health is directly correlated to our health, and not just physical, which is, again, the focus of these food webs, but spiritual, mental, economic health. So again, we are actually cleansing the landscape through fire, not just the physical reduction of pests and disease, but also spiritually, smoke has important cleansing effects. And really fire, indigenous fire, is the core of traditional agriculture. So often we hear Native Californians referred to as hunter-gatherers, and to me that term is actually does tremendous disrespect to large-scale ecosystem management that existed in this state and continues to exist. Spiritual benefits, as I mentioned, we're really trying to create careers that provide a place for ceremony and cultural revitalization. It's so hard in this day and age for tribal members to actually find a pathway where they can provide for their families and communities and actually not sacrifice their culture and their identity. Social benefits, this is an intertribal crew, so a big way we're actually building the capacity for this paradigm shift is intertribal collaboration. One tribe obviously cannot tend to our entire ancestral territory due to the effects of genocide, land loss, and so many other things. So it's really about bringing back this intertribal collaboration and rebuilding our community. Economic benefits, as I mentioned, we're trying to decolonize careers and we're trying to build the capacity for a paradigm shift that we can actually sustain. And I'll dive more into ecological benefits in these following slides, but just a little bit on the dichotomy between cultural fire and prescribed fire. Obviously, prescribed fire is much more recently introduced. It's heavily influenced by this sort of paramilitary ideology. You're fighting fire, suppressing fire. Cultural fire sees fire as a relative, a gift from creator, and a tool that's been given to indigenous people, as I mentioned, to provide a foundation for ecosystem management and ultimately large-scale agriculture. And going back to my previous point, when we look at oak woodlands in California, you see the strategic timing of cultural burning and the duff layer around these oak trees to target the two main pests of acorns, the filbert weevil and the filbert moth. And to me, this has all the elements of conventional agriculture. The only reason that we have a hard time making that parallel is because it's so much more sustainable. We still see emphasis on nutrient cycling, fertilization, pest and disease management, yield. All these things are important to indigenous agriculture. We just don't see the implements of diesel, tractor, massive land changes, and so it's hard to associate it with what we typically think of as agriculture. And again, most prescribed fire is really focused on acres treated, acres black. There's often very few cultural objectives and few ecological objectives. It's the opposite for us. When we're burning as a tribe, we have a long list of cultural and ecological objectives. And lo and behold, at the end of the day, we've accomplished fuel reduction as well, but we've also accomplished a lot more. This is a great quote. This just speaks to the fact that this is not a new conversation, not a new problem that we're having. This was a letter that was written by Klamath Jack and sent to the Fish and Game Commission in 1916, explaining the importance of cultural fire on the landscape and the effects of fire suppression. I don't have time to read this, unfortunately, but I do encourage everyone to check out this website, Karuk Tribe Climate Change Projects.com. There's a lot of great information on there that can further your understanding of indigenous ecosystem management and the history of cultural fire suppression. Actually, some of the first laws in California were passed to suppress indigenous ecosystem management. As we know, when you dismantle a food system or a food web, you dismantle those higher trophic levels. And of course, that was an unfortunate and intentional aspect of colonization. So why burn? Increased quality and abundance of culturally significant plant materials. This is a deer grass. The lower image shows right after the burn. The upper image shows the regrowth and the increased abundance and quality of flower stalks, which are a basketry material. We're reducing pest and disease populations. We're also allowing sunlight to actually reach the meristems of this plant. The deer grass growth becomes very decadent with time. And so just like Thule, allowing fire to provide a pruning mechanism for these plants is actually extremely important. And we're maintaining heterogeneity and species diversity because cultural fire is not acres black. We're creating a patchwork, a mosaic of disturbance on the landscape, and that's contributing significantly to spatial and temporal heterogeneity. which I wish I had more time to go into. It's a lot to cover in 15 minutes. Cycling nutrients, as I mentioned, we're greatly increasing carbon sequestration when we're burning, which is counterintuitive to most people's understanding of fire, because we see these massive carbon releases during catastrophic wildfire events. Actually, the opposite is occurring with sustainable fire-based forest management, where we're selecting towards larger, longer living trees that ultimately result in more net carbon sequestered. That's something that hasn't been studied very much. So again, I'm really grateful to be here, surrounded by so many amazing researchers and academics. I would love to find ways to collaborate more on how we can bring the Indigenous voice into literature and explore some of these hypotheses around the importance of cultural fire management for Delta restoration. And I really do believe that this is a core element of revitalizing the Delta that is not being emphasized or discussed as much as it should be. Wow, that was a quick 15 minutes. So a little bit on the different morphological changes when you burn redbud, for example, which is a very important basketry plant in Northern California. This is a great study by Jonathan Long and Frank Lake on burning in black oak woodlands, showing a little bit about these food webs, right? But from that cultural perspective, and delving into some of those interactions that are often missed when we don't engage with cultural practitioners when looking at these food webs. This is the work created with SFEI. It's a collaboration between Shingle Springs, UAIC, and the San Francisco Estuary Institute. This is sort of our draft white paper that I mentioned yesterday, trying to show this spatial and temporal scale of cultural fire management and gathering, which is also an important secondary layer of temporal and spatial heterogeneity when the post-fire gathering takes place. So as you can see, reduction in dead growth, the tule biomass and productivity greatly benefits from burning because it has no self-pruning mechanism, and there's no way for the new growth to easily come up with all of that dead biomass in place. Tule shoots are also an important food source. So being able to go out and gather tule shoots and not having to bushwhack is an important thing. Here you go, month and a half, or rather just less than a month post-fire. You can see before and after just how vigorously that tule has grown back. And you can also see the heterogeneity that has helped to create. Before the burn, this was all dead, overgrown biomass. And now you can see there's a tremendous amount of new growth, but there's also some patchiness where that old growth, some of that decadent old tule and cattail remains. And this is highly economic. If you were to try and do treatment methods with modern restoration approaches, you'd be spending a lot of money. This is something one person with a drip torch and a couple people with a garden hose can do. Don't think I have time to go into this too much, but this is just a point about how the importance of emergent vegetation really factors into fish diets. This is research from Mr. Young, who I think might be here today. We have obviously a large portion of the diet coming from emergent vegetation, which is why it's important that we're focusing on how we're managing these terrestrial ecosystems, because they do directly impact fish diets and survival rates. So just really want to emphasize, yes, I'm talking a lot about emergent vegetation. I know this is mostly supposed to be focused on purely aquatic resources, but fire plays an important role in aquatic systems as well. I don't have time to go into the changes in pH and water quality from cultural burning, but again, opposite from what you'd experience in a catastrophic wildfire. You see benefits to water quality. There's an example of that patchwork, that island of green tule in the center burned. Their surrounding cattails did not experience any fire. And so we're doing tremendous amount to support the temporal and spatial heterogeneity. And there's significant physical, chemical, and biological benefits. I mean, you can imagine that increased light penetration has benefits for lower trophic levels like phytoplankton, and of course, invertebrates that use tule as a food source. You're also opening up this patchwork. So sedges, forbs, plants that would be quickly outcompeted by tule can actually have a moment to have their time in the spotlight. Way over time here. Great book, Kat Anderson, you probably all know it, but it really speaks to the scale of indigenous ecosystem management cannot be overstated. Hundreds of thousands of people, hundreds of languages existed in California pre-contact. And I highly recommend reading this book if you are not aware of the role indigenous peoples have played in shaping the ecology of this state. Don't have time to talk about this. You can see invasive species on the left, native grasses on the right. Only difference here is strategically timed cultural fire management. Many restoration practitioners would spend thousands of dollars using selective herbicide in order to get these kinds of effects. You could do this on a budget, and it also is unlocking many synergistic effects that fire has, such as increasing the fertility of the soil, enhanced organic matter, things of that nature. Just a little bit about alliance building, which I don't have time to go into. Oh, didn't mean to do that. And yeah, any burning questions? I guess we'll wrap it up there. There's my emails.
Thanks, Zachary. We can take one question while Edmund is loading the next slide. Ryan, go ahead. Don't fight about it.
It's Ryan Bellmore. Wow, it was such a program. It's really just exciting to see this. And yeah, congratulations on getting something like this going. You mentioned sort of this novel ecosystem component at the beginning of your talk, and made me wonder, there's probably now, I know like invasive species have changed fire dynamics in a lot of landscapes, and like, are you having to deal with that here as well?
Aquatic systems, fire does have limitations. Obviously, submerged aquatic vegetation is not something you can treat with fire. But yeah, we have seen, so for example, in the mixed conifer oak woodlands that we're doing most of our burning in the Sierra foothills, we have a tremendous amount of annual grass crop that has come in, and that does change the phenology of when we're able to burn. What I've found for strategic timing of invasive species management, like I had shown in that one picture, is that in some ways the contiguous annual grass crop can be a benefit because you can time your fire, because those are 1 hour to 10-hour fuels, right? They have a pretty short lag time. So you can actually, in native grasses, if they're still present in those meadows, if you have strategic timing of fire, when those annual grasses have just germinated, you can actually get them at that stage of their highest vulnerability, and that cured grass crop from the previous year can assist with bringing fire through that system. So in some cases, it gives us more prep work, and we have to invest a tremendous amount of time in order to burn some of our units. But in other cases, that annual grass crop has actually made it easier to put fire on the ground through sort of a larger burn window than we would have typically have. So long story short, there's trade-offs. But in general, yeah, invasive species have made it more difficult. And there's some species that you have to use an integrated pest management approach, like Himalayan blackberry, for example, in our riparian areas. It's very hard to burn it on a consistent basis, right? Because you lose that contiguous fuel bed as soon as you burn year one. And so bringing in goats or grazers or something like that could be very helpful. But really, one of the main takeaways I think for land managers here is fire is a tool many of us aren't even considering using on the landscape. So if we could at least bring fire back into the fold, it would make a tremendous difference.
Our next speaker is Andre Buchheister. Thanks.
Well, good morning. For those of you that were here yesterday, this is kind of an extension of my talk yesterday, but I was asked to provide a little bit of the nuts and bolts and details on Ecopath with Ecosim, or EWE. And so this is kind of a crash course or introduction into the modeling method, getting into more of the details. So as you know, there's lots of different ecosystem modeling platforms available out there, but Ecopath with Ecosim is certainly the most common for fisheries applications. This is an old map here, but you can see throughout the globe, we've had hundreds and hundreds of ecosystem models developed in a variety of different contexts. So EWE really has three main components. I'll focus on the first two, Ecopath and Ecosim, and then a later talk will go into more detail with Ecospace, but I'll at least introduce it here. In terms of the software packages that you would use, the user interfaces are really easy. They're free, open source. There's a Ecopath software that you can download, but it's also been implemented within R for facilitating higher numbers of simulations and manipulation. That's a package RPath. So Ecopath, as we were talking about yesterday, is really just a biomass accounting tool for the ecosystem. It provides a mass balance snapshot of the system. This is a figure that I showed you yesterday for the Northwest Atlantic Continental Shelf model that I helped develop. And you can see the complexity, right? Ryan was talking about ranges of complexity. This is on the higher end of trophic complexity. Those nodes represent the trophic groups where size is relative to biomass. The model estimates what the trophic level is for the different groups based on diets, and we can quantify those linkages and map those out. And we also have fisheries behind the scenes here that I haven't depicted. So in terms of the theory and the equations behind Ecopath, it's really based on two primary master equations, the production and consumption equations, and you can think of it as different pie charts, if you will. So all the consumption that a predator consumes, right? That biomass can be converted into production, respiration, and unassimilated food. So we can account for that within in the pie, right? Once we remove respiration, unassimilated food, what's left is production. And that production can go towards harvest or catch, predation, other mortality. And we can also build in net migration or biomass accumulation through time. And so all the different groups are linked in the food web in this fashion. I don't have the scarier equations. I didn't want to do that in the morning. So for Ecosim, this is now the time dynamic version of the model. So we're now seeing how biomasses, catch rates, et cetera, change through time. So within the model, what's driving the changes in the food web and in the ecosystem is typically going to be fishing mortality or fishing effort in these types of models. And then we fit those predictions from the model to time series of relative biomass and time series of catch, as you see here in this example, where the points are the observed time series, and then the red lines are the predictions for a number of different groups within the model. And a common trend is that you'll fit some time series well, and some time series you won't do so well. And a lot of times that's tied to how reliable the data are. But this is, a way of trying to find the best fit to the available data that you have, and some tweaks need to be made. And this is all done automatically by minimizing sums of squares. In terms of the equations for Ecopath, for Ecosim, it's just time dynamic versions of these equations that I have on the board. And you set up a system of differential equations that the computer solves for. One of the key components within Ecosim is how to model changes in consumption. And that's done using something called foraging arena theory, where the prey biomass is split into two different pools. There's the unavailable prey or not vulnerable prey, and the available or vulnerable prey. And so there's a vulnerability parameter that regulates the transfer rate between these two biomass pools, and predators can consume them when they're available, when they're outside of some hypothetical refuge, if you will. So these vulnerability parameters ultimately end up being a knob that controls the relative bottom-up or top-down effects for a given predator-prey interaction, and it regulates the biomass gains that a predator can have given the prey. And as I mentioned, these are the parameters that are largely estimated and fit to match those observed time series. EcoSpace is this third module. It's the dynamic spatial version. In essence, you set up a raster or grid system and you run an EWE model within each of those, and then you connect those cells with movement, with connectivity. You can lay different habitat preferences, but Kim will talk a little bit more about that coming up here. So I want to emphasize one of the things that Ryan was saying. We really do want to tailor these models to the specific objectives and available data, and that results in differing levels of taxonomic aggregation. This is the aggregation and setup that I had for the NWAX model. You can see a lot more aggregation at those lower trophic levels, as well as the higher trophic levels with the apex predators. And then we start to tease things out in more detail for some of the forage fishes and for the fishes. The reason for this was because we were interested in Atlantic menhaden and some of their key predators. So we wanted that level of taxonomic resolution. We can take individual groups or species and we can break them out into different age classes to account for ontogenetic shifts in diets. We can account for selective harvest by the fishery. These do require some extra growth and mortality parameters, but it's typically not too big of a problem. Now, for those of you familiar with single species stock assessment, we're not using any stock recruitment relationships in this model. It becomes an emergent property from the way that you've set it up from those vulnerability parameters. And then specifying the structure of the fishery fleet, lots of flexibility there. You might want to define your fleets by gear, target species, whether it's recreational, commercial. You can account for bycatch. These are all things that you can build in. So what about the core data needs for Ecopath? In bold here are the four primary parameters that you need for each group. And you actually only need to enter 3 of them, and then the 4th one is estimated by the model. So production to biomass ratios, consumption to biomass ratios. These are things that we often get from literature. There are empirical methods to estimate these. You can also get them from stock assessments. Ecotrophic efficiency is the proportion of the production that is used within the system. That's typically what we estimate within these models, but not always. And then undergirding that, we have a diet composition matrix. That's going to come from literature, from surveys, et cetera. And I did want to mention that you can allow for some import of diet from outside the system, and that's useful if you have migrating mobile species that are gaining some of their consumption from outside the system. We account for the fraction of consumption that's not assimilated, and we also want any annual catch estimates for the fished groups, and those will come from stock assessments or landings databases. There's certainly other data needs. I'm not going to go through all the detail here, particularly as you shift to that ecosim. You're going to need time series of different things. So time series of relative biomass, time series of catch, fishing mortality rate, fishing effort. For those age stanzas, you're going to need some total instantaneous mortality rates for each age group, von Bertalanffy growth parameters, et cetera. And so there's also other data streams that you can build in, and I'll mention a few of these later. In terms of data sources, a lot of these are going to come from stock assessments, from available survey data, or from the literature. Kind of do your best to find what's out there. One of the nice things is that you can build in these forcing functions into the model that modify the parameters through time. So I should have mentioned that typically EWE models are annual, but behind the scenes, they're calculating things on a monthly time step. And so we can have monthly or annual forcing on any of the parameters. So this can be useful for evaluating bottom-up effects, climate change impacts, eutrophication, seasonality. So here are a couple examples on the upper graph on the right. This is work that a graduate student of mine did by adding in a primary production time series and then using that to forecast forward different scenarios that are predicted in the literature for declines in primary production resulting from climate change. So there are estimates of 3.5% decreases or 6% decreases in primary production. And so we can simulate that through time. We can also look at seasonal changes. The bottom graph here is an example of seasonal forcing on those predator-prey vulnerability parameters. This is some explorations that Dave Shigaris, a collaborator, did, trying to see if he could better model striped bass diet on Atlantic herring. So instead of being consistent through time, these diet proportions are now changing seasonally, which are the blue lines there, just to account for changes in predator-prey overlap. Obviously, as we were talking about yesterday, one of the big advantages is exploring scenarios and different management policies. And typically these are user-defined scenarios. So your what-if scenarios, that's going to be case specific for whatever system or study you're interested in. EWE also has these open loop policy explorations where you can run simulations that optimize 4 weighted policy objectives. So you specify the weights of these 4 things. I haven't done this. I go with a user specified approach, but you can maximize fisheries profits. So that's nice to mention that you can build in some economics into this. You can maximize employment. You can maximize rebuilding of species, maximize ecosystem structure or health. There are specific quantitative measures for each of these, and you can optimize the fishing industry to achieve those goals. Addressing uncertainty is always something that we want to be cognizant of. One of the approaches to do this is to indicate the relative level of uncertainty for different parameter inputs. These are known as pedigrees. This little table here just represents different parameters for different groups. And the color code represents how confident are we in those estimates. So red, we have high confidence. We think there's a lot of precision in those estimates. And then blue, man, we don't really know what's going on. There's a lot more uncertainty in those parameter estimates. And that can feed into some Monte Carlo simulations where you run the model, where you resample the parameters from these distributions that are determined by our level of confidence within those parameters. And that's this bottom plot here just shows predictions for relative biomass and relative yield for different groups, and we can start building in some uncertainty thresholds. And you can certainly do some sensitivity runs of your model with alternative parameterizations. If you're not really sure what the natural mortality rate is for some group, maybe you have a model where you go with one assumption and another model where you go with another and compare those. A lot of work has been done in trying to standardize EWE modeling methods. And so there's some great guidelines available for model development. Here are 3 papers to highlight. And so there's lots of detail in here, but maybe one big picture takeaway is that we do want to tailor these models to the specific goals, to the specific systems, specific questions. Sometimes there's a lot of parameter borrowing across models, or taking a model that was developed for one reason and then applying it a different way. We want to do our best to avoid that and have it developed from the ground up for the purposes that we have. Wow, apparently I'm flying through this. I did want to highlight some pros and cons of EWE. As with any modeling endeavor, you have advantages and disadvantages. You know, again, these are some things that Ryan was talking about, which I think he did a great job on. So I'll just highlight a few here. So first, synthesizing information and quantifying the system. Just providing that description of a food web, of an ecosystem is really valuable so that we can start to better understand the system. Now, the downside to that is these types of models are very data intensive. That also increases the amount of uncertainty in these models. So there's always these trade-offs in model resolution and scope and scale. It's a great way of identifying gaps in knowledge and in our data, right? As you're parameterizing these models, you realize, wow, we don't know very much about these interactions here. Or we need more diet information for these groups. Which are the key players in the system? There's the process of building these models will identify those things and can help prioritize future research. The downsides are that you're going to come across those data poor groups and you're going to have to deal with them. Also, some of the parameters that are used within the model aren't things that you can empirically measure. Things like those vulnerability parameters, for example. A vast number of indicators can come from these models, both from kind of food web ecology context, but also from a management context. We do see that sometimes those indicators can be sensitive to different parameterizations. That also leads to some skepticism by folks. Man, this model is way too complex. I'm not going to put any stock in it, right? But again, it's all about finding that balance. You know, the box quote there, all models are wrong, some are useful. We got to find that sweet spot there. So making and evaluating hypotheses. We can't run experiments on entire ecosystems, right? We don't have the replication, and we certainly don't want to mess things up. So these digital experiments are a great way to test some hypotheses that you have and evaluate what some potential outcomes would be. This speaks to a comment made yesterday, right? The importance of hypothesis-driven research. But again, we have to highlight the downsides of uncertainties in the model and in prediction. There was talk of non-linearities, unknown futures, extrapolation, et cetera. Those are all caveats as well. So these approaches are great for for management strategy evaluation, policy exploration. I gave some examples yesterday. There was different case studies that talked about that. Challenges, how do we integrate outputs from these types of models into existing management structures? I'm in the fisheries world, so we have single species management, and trying to build in these ecosystem considerations can be a challenge. And there has to be shifts in changing that, not only in the thought, but also in the systems. And then evaluating trade-offs. That's one of the themes I try to point out to my students is there's no black and white answer. There's always kind of pros and cons to different management solutions and different approaches. Typically, EWE models are often used in a more strategic sense, given some of the challenges. What directions do we want to head? What are solutions that tend to move us in the right direction? Typically is not used for tactical management, but that's changing a bit, and the Menhaden example is a good example. So with that, I guess maybe some concluding thoughts here would be we want to take advantage of those pros and try to minimize those cons, and it's all going to be dependent on what those questions are. But it can be a really useful tool, and I'm excited that you guys are exploring that for the Delta. Thanks.
We have time for questions. Go ahead. Steve.
Yeah, Steve Lindley, No Fisheries. Andre, thanks for that great overview of Ecopath and Ecosim. And I think you were getting at this with your last slide there, but just I wonder if you can delve a little more into the issue that Ecosim, taken at face value, say if we make a change in the management of the system, it'll quickly come to a new equilibrium and there will not be much or any variation. And real ecosystems never do that, right? They're super variable. And you've even worked on Menhaden, so like coastal pelagic species are characterized by boom and bust cycles that are mysterious in their origin. So I'm wondering, what, given that kind of disconnect, what are the limits to this kind of approach in helping managers develop resilient strategies?
Yeah, no, that's a great question. And that's where I think the combination of approaches is really useful, right? So building in some recruitment stochasticity within single species models is a way to address some of those challenges. And that can be good for the tactical side of things. Using these broader models for strategic planning can be really useful in terms of kind of the directions that we go. As work continues, I think it's also possible to start to build those types of things in, right? With those forcing functions, we can change and build in some stochasticity and changes through time. So again, it's finding that balance between is it simple and good enough versus how realistic do we want to make it? So there's that tension there. And I haven't delved too much in those realms, but I think that's definitely a question that a lot of folks are tackling.
Diane McKnight, Delta Independent Science Board. I was wondering about how these models might be used to quantify ecosystem resilience, ability to recover to a new state or a previous state. As highlighted in this poster, we have HABs, fish kills happen. You mentioned you can't do experiments, but we have some experiments happening. And how would these models be used to quantify resilience if that's one of the management goals?
Yeah, I think I'm going to build in another comment from yesterday too. This idea of different alternative stable states is one of the challenges, right? As you tweak and modify things, it's going to kind of settle on a solution. But if you start having lots of changes to the system, there may be some alternative stable states that we're unfamiliar with, right? And so that's a challenge. I don't know of, and I have been up with the literature in the last few years, how much people are exploring that kind of idea. But yeah, in terms of exploring those types of stressors and challenges, I think that's where it's nice to set up different models or scenarios that address those specific hypotheses, right? Where you think maybe some of those linkages are going to be changed, or the strength of those relationships might be changed. So you can set up a model with one kind of hypothesis and scenario and another one, and you can simulate the impacts of some HAB or those types of things.
Kenny Rose, Delta ISB. So I'm always very skeptical of off-the-shelf models, right? And we should be. So there may be people out there who, with the Delta, don't think foraging arena theory applies to certain interactions. Or if you go to ecospace, that you can't flux fish like they're nitrogen to simulate their movement patterns, right? So how much have you gone in and modified the source code to accommodate, as you go from strategic to tactical, those kind of details become more and more important? Have you worked with the source code itself, or are we going to inherit foraging arena theory forever? Or that's what I'm always a little worried about with the off-the-shelf models.
Yeah, that's a fair concern. The answer is easy. No, I haven't dug deeper into that. And for a little bit more background on the foraging arena theory, it ends up being more like a type 2 functional response relationships. You can build in some predator prey switching and making it more of like a type 3 functional response. So there are some ways to kind of modify the parameterizations within that to achieve different types of behaviors that you may want. But yeah, those are some kind of simple thoughts. And in terms of the off the shelf, again, it's all about these trade-offs, right? Building something from the ground up that is super specific and fine-tuned versus something that's good enough in general. Those are always the tensions at play.
Okay, I think one last question. Fred, did you have one?
Fred Fira, USGS. I guess kind of extending off of Kenny's question, I was just wondering, so you showed early in your talk those plots of kind of this ubiquitous issue, right? Where the model works well for some species and not for others. Oftentimes that's a data issue, right? But I'm wondering for people like you and maybe Ryan and Steve and maybe even Kenny, people who do this for a living and are modeling practitioners, have you noticed any kind of generalities in terms of, aside from the data issue, what might be contributing to some of these things? Like, are there particular life history strategies, ecological aspects of species that seem to contribute more to some of these uncertain outcomes than others?
Yeah, interesting question. Yeah, a lot of the, most of the stuff that I've seen has been more on the data side, right? We don't know very much about a specific group, or we're estimating time series from a trawl survey, which is not well geared for surveying some specific species. So most of the focus has been on that side of things for me personally, in terms of whether there are some commonalities in terms of ecological or life history characteristics that tend to indicate poorer fits in those. I'm not as familiar with that, but it's an interesting idea. Yeah.
Okay, thanks, Andre. We'll move to our next speaker, Kim DeMutzer.
Good morning. I was hoping this would play on its own. So there's the little video behind us that is not just to distract you from what I'm trying to say here. This talk is about advanced models or advanced modules in Ecopath with Ecosim. I'll mostly be talking about ecospace. I've learned there's an interest here in contaminants as well, so I'll talk a little bit about ecotracer as well. But what you're looking at here is including video games to ecospace output. So perhaps in outreach or even in talking to managers, instead of looking at ugly looking maps or squiggly lines, you actually want to look at fish that are swimming and they're increasing, and you can do split screens where you have one management action on one end and one on the other, and models of the species that are actually occurring in your system, and when their population grows, you'll see more of them on the screen. So there's some thoughts and developments in changing the visualization of this model output, and this is one of those things that we're looking at right now. All right, so why do we want to venture into this area of creating these complex ecosystem models? I think in trying different management strategies, even though there are a lot of challenges in replicating that system on the computer, you are better off if you can try things out on a digital twin. So I think that really is the idea of creating a digital twin of your system and trying out these different management ideas on the computer first before you start doing it in real life. So ecospace is not going to predict the future, right? So if that's what, that would just be a wrong expectation. But you may be able to look at what would change if we implement something versus if we don't do it, and look at those differences. So we planned it so that that I don't have to talk about all of this because it wouldn't fit in the time. So you've just learned a lot about Ecopath, Ecosim, and a bit of an introduction Ecospace. So I'll talk a little bit more of what you need if you want to develop an Ecospace model. And some additional modules are there as well. You can actually create your own plugins if you want to look at different things. And if it works well, it becomes part of the source code. And so it's really an evolving type of model that you can use. But again, I'll just touch on two really. So EcoTracer is one module within this software. So you always start, I would say, with building an Ecopath model. You want to calibrate an Ecosim, but then maybe you want to start doing some different things. How do contaminants flow through a food web? What I'm showing here is actually just an example of how it's used. So a colleague of mine, Kieran created an EcoTracer model in Scotland and focused on radioactive carbon, but it could be something else as well that you're tracing through the ecosystem. So if you kind of think of you know, the kind of the inputs and outputs setup of an Ecopath model, you can imagine you may be able to add some contaminant tracing to that. So you have contaminants that exist in these different compartments in the environment and in your different groups in the model or your species. And you have Ecopath and Ecosim kind of working in the background, and the trophic interactions that you've indicated in your diet matrix really form the basics of how contaminants can flow through the ecosystem. So this is really contaminant flow as it would spread through a system through trophic interactions. So there's transfers. So you have to, so here's a list of inputs again as well, right? You have to know the concentration of your contaminants in the environment. And then there's uptake by the species. So you are actually defining that uptake rate. But then your predation is defined by your connections in your Ecopath model. And then you can have immigration and emigration of species in the model. So if there's contaminants in there, that would also be a way of how the contaminants move in and out of the system. Contaminants can be removed from the system. Either it flows out of the system, environmental outflow. There's excretion from the groups in your model, and mortality of the groups that would take that out. So here again, the example of when this was used for radiocarbon. So you do want to. So the things you need to know is that inflow of whatever your contaminant is in your system. What is the concentration in your environment? These are just two groups, so a consumer and a predator. And of course, in your food web, you have all these different connections. What is of your group? So this could be a species, right, or an aggregate. What is that absorption of your environmental contaminants? And then there's some physical decay that you need to know. There's also assimilation that happens that you'll need to know. Then there's a portion that's excreted. This whole group can die. That would be the mortality. The rest, you know, is basically contained in this group and then becomes available to a group that consumes on that group. And then those consumption rates are all determined in your Ecopath model. So that would be determining the flow. So there's dietary uptake of contaminants as well. So with that, again, I think with a lot of applications of Ecopath, you need to know a lot of the inputs from either field measurements, lab observations, what it really does for you. If you know at least these type of rates, is how then would that spread through the ecosystems? Which species would have certain levels of those contaminants? So it kind of just helps you with where is it flowing to if we have some type of incident and we have this much of a concentration of some contaminant in the field here. So just, I would say a new module, but just a recently more used module where more people are looking at it, and with kind of this consortium-based freeware type software that Ecopath really is, the more things are used, the more they are improved, and there's been a lot of recent changes in how well this functions. So it allows you to quickly model contaminants, flow and accumulation in ecosystems, different compartments in your ecosystem, food web. And there's been some great examples of use by now. There's currently a project underway looking at microplastics. So again, it just like, where does it end up if we have these type of food web connections is what you can really get out of it. All right, where I spend a lot of my time in this software is using it for EcoSpace. And again, because you just learned about Ecopath and Ecosim, I'll dive right in, where really all you add now is a spatial grid. So I think in problems like restoration and changing things in the environment, this is probably where you would be interested in having a spatial resolution of your model, just because there's spatial heterogeneity in environmental variables, or you're changing something only in one part of your system and you want to be able to include that. So EcoSpace or Ecosim is really running behind the scenes. But now we have cells, and the organisms can actually move between the cells. Fishing fleets also actually move over your model area. I would say it may sound more complex than it is, because that's where a lot of the movement in space is really located. The other changes in space, so you have a different environment through time, right, are maps that you read in, so preferably from other models. So each time step, your environment changes, not because we model all those complex changes that happen if you have freshwater inflow somewhere, what would the salinity be here? I'll have my hydrodynamic modeling colleagues do that, and I'll read in every month a map of what the salinity is in each of the cells. So that's really how the species can respond to these spatial changes. It's really output from other models, and the only movement is the organisms themselves and the fleets that fish on them. You can incorporate advection, though, if you think it will also affect your organisms. So it will be part of your hydrodynamic models. If you think that's enough, then that's it. But if you think, well, they're moving because they're dispersing, but they're all kind of, pushed to the east because this is going on, you want to incorporate that explicitly in your model. So there's just by including that information for other models, now you can have spatial variation in productivity. Perhaps it's more fishing you're interested in, so spatial variation in cost of fishing. And I would say Ecopath was really designed to look at fisheries questions. So EcoSpace started to look at MPAs. That's really a spatial fisheries management practice, right? Looking at marine protected areas and how that would affect your landings or your cost of fishing or anything like that. And it really recently got expanded to, well, if we have this spatial area, we can look at how suitable the different areas within your model domain is, and especially if we start changing things, how those suitabilities change. So you can represent these habitat preferences by group. And you can actually sometimes, your fish don't just move in response to going to more suitable areas. Sometimes they have some type of migration patterns. If that's the case, you can actually include that actively in your model. So if you are interested in ecospace, you would still construct your Ecopath model. So that is what I call that digital twin. Like you want to first replicate your model on your computer. We still use Ecosim. It's still our best calibration methods to look at observations through time and fit your model to that. But then you want to create a base map, maybe read something in. You always include a map that includes at least bathymetry. That's kind of what you start with. You choose your grid size. I wouldn't make it too small. Think about Ecosim running in each of these cells. So your trophic interactions need to be able to occur in a cell of your model. So usually I don't go a lot smaller than a kilometer square, and some have much bigger cells. So again, those type of things depend on what questions you have. Ecospace is just a different way to looking at it, right? So think about what your research questions are. Are they spatial? It's not necessarily better. It's just that it adds space as a component. If you think you need that, well, maybe you want to create an ecospace model. If not, maybe Ecosim would be where your end goal is. And then conversely, I also would like to add again that I don't think Ecospace is necessarily more complicated than Ecosim. You can create a pretty simple model with not a whole lot of groups in there, but you happen to be very interested in a spatial question. So then, go to Ecospace to answer that. So what do you need? Again, that base map you need, that's really gives you depth, so a bathymetry map. You can now, we already talked, or Andre did, on the primary productivity as a driver in your system. You can have that as a spatial map as well. So that would be an optional map to include. The excluded cells, I think that's a very useful item. Your ecospace model area is usually a square of some kind. But now you want to receive output from another model. So you want to really make sure that each of your cells receive output from that other model. So with an exclusion layer, you can make your model area exactly like the model area of your companion modeler so that you don't have cells in your model that don't receive input from another model. So you then can include habitats or environmental drivers that are spatially explicit, so these environmental conditions. You can also, again, think of it, I'm interested in a spatial component because there may be spatial fishing closures in my area. Sailing costs can be included by including ports and how far these fleets need to go away from them to catch fish. And I see Steve standing already, so I'll go a little quicker. Here's a quick example of when you're interested in, well, certain areas you won't use certain gears of fishing. So that's a good question to address with Ecospace, because you can indicate where these fleets can go when they fish. You maybe want to test again what the effect is of an MPA. A certain area in your model area is closed for fishing, and this could be closed at certain times a year and not all. You can indicate all of that. I think most of this I mentioned. This, I think, is an interesting kind of new addition of how to do this, what I mentioned already. There's a spatial temporal framework that allows you to maybe save a folder on your computer that has included the output on per month of another model as maps, really. And you create a connection that allows your ecospace model to read in a map every month of every parameter that you're interested in, or maybe you want to read in some only every year or every 5 years. It will keep the information from your last map unless you tell it to read in a new one. So that is really, I think, a new advance in ecospace to kind of really work in this way and rely on other models' outputs to create your environment. Then, of course, your species need to respond to these drivers, so you create different environmental preference functions for each of the environmental parameters that you read in. One quick note is that that spatial temporal framework is really only available through a pro license. So there's freeware, and you just cannot do all the things in the freeware. It's with the pro license, you get troubleshooting help from Ecopath programmers as well. So there's a lot of back and forth, I can tell you, where if you are not the one changing the source code, but you want a change because it's not doing something that you wanted to do, through those interactions with a pro license, they will help you do it. So here's just an example: primary production, temperature, habitats. It kind of all works in the same way, creating, I would say, more and less suitable cells for each of your different groups in the model. And then here is just a map of different, I guess it's dynamic, models that are used for management. So it's getting more and more used to answer specific management questions. I don't think a lot of people realize what advances have been made in ecospace because there's no user manual anymore that goes that far. But it's in the work, including a textbook. Right now, though, I just published this year a book chapter that includes all those advances with a long list of collaborators. So there's a lot of case studies in there, which you can get to it with this QR code. But email me because it's a book chapter that's not open access, and I can send it to you if you're interested. With that, I will take any questions if I am allowed.
Yeah. Thanks, Kim. I feel like a Grinch up here, cutting off all these excellent talks and cutting the discussion. Clearly, the organizers should have planned for 3 days, and I'm one of the organizers. Any questions for Kim? Yes, Lillian.
Lillian McCormick, I'm a postdoc. So Kim, really interesting talk. I was wondering, based on something that came up yesterday with the EcoTracer module, we saw yesterday that contaminants passing from maternal transfer to the eggs may be really important, and I was wondering whether EcoTracer has the capacity to do that right now, or will maybe in the future.
I don't think so, really. So what you can do, it's not really set up for that. I would say you could see if you can include that as linked groups, but you will have to think about that. It's not truly meant for that. Really, the kind of the linked groups are more of like the life stages rather than eggs. You could make, a group out of eggs if you just see it as food, perhaps, and then where that's the contaminants in that, where those are going through the web. But yeah, it does take some kind of artificial maneuvering because eggs aren't actually in the model.
Okay, one question in the back.
Thank you. Sean Acuna, Metropolitan Water District. I was wondering how this works with how does this compare to a vein transport model, like the SWAT model that's been used for tracing the vein transport of contaminants. This seems to be more integrating more of the biotic side, but the SWAT model is able to integrate very well with hydrodynamic models. I was wondering how this compares.
Yeah, I don't really truly know the answer to that question. I would say what this does here is tracking it through the food web by looking at who eats who, what are the decay rate, what are the assimilation rates, what is left behind and what then goes to the next group if it gets consumed at a certain rate. So it's really the flow within the food web only. And it sounds like what you're talking about is more in the environment. I would say this would be a perfect example of where you would maybe use that model to get at what is that initial concentration in the environment and how does that change through time, which you can then read in. You can use EcoTracer in EcoSpace so that your environment, for that to be variable, you'll need information from outside. So I would say it's more of a model coupling than a model comparing issue.
I'm afraid we have to move on. And our last speaker before coffee is Dave Beauchamp.
Thanks, Steve. I'm going to chat about bioenergetics models, and you've heard a lot about different scales of modeling. My own comfort zone happens to be I like to understand kind of the tactical, mechanistically driven interactions that can then inform how these more strategic models operate. And so this can provide a lot of the underpinnings of that. And so some of the things that are appealing to me are that bioenergetics models allow us to look at relevant spatial and temporal scales and focal species and even the life stages within those species that are really timely for management in the metrics that are useful to managers, both in terms of resource management, but also in terms of restoration and recovery and conservation. We're going to talk a bit about how thermal effects really influence species distributions and thus the overlap among species or among life stages and how that can really facilitate food web interactions overall. Spatial temporal patterns in the energetic growth potential. I think you probably heard yesterday quite a bit about spatially explicit modeling. This is some of the underpinnings of that, in that we can look at the net energy benefit that's provided by different habitats within a greater ecosystem, and what's the connectivity there, which then gives us some information about life history strategies and the diversity of life history strategies, which may be successful under different types of conditions. And some of those conditions can be altered by resource management, and others we just have to take the hit, but understand where that's coming from. And so that can be very helpful in that regard as well. And then from quantifying these food web interactions in general, it's often important to know what's the seasonal carrying capacity. You may have lots of food available, but it has to be available when it's needed. And so looking at that through these critical growth periods, for instance, or periods of critical interactions, we've got the temporal and spatial resolution where we can address some of those questions a little more directly. And whether it's just seasonal carrying capacity within a species or that's exacerbated by competition with other species can be important. And then if we go up a trophic level, then what's the predation mortality that can be imposed by some species on others, and what are the underpinnings of that? What size? What are the species that are the main predators? What sizes of those predators? What seasons? Maybe even what locations those critical interactions are happening. Can be addressed at a little higher resolution based on a bioenergetics approach. And then we can use these sorts of approaches to then examine the potential impacts of invasions of species or climate change, or the feasibility of introducing species, say above dams or in other places. We can do the thought experiment before investing millions or billions of dollars in that type of thing. I already talked about climate change and water operations. This all plays through. And then you just heard also that since a lot of bioaccumulation of contaminants is happening trophically, because we're able to estimate consumption by prey type through time, that gives us again a higher resolution map of how bioaccumulation might occur through the food web. And then there's also ways to look at nutrient recycling too, which can be an important way of transporting nutrients to otherwise nutrient poor environments. So some of the common questions that require some information on trophic interactions are what's limiting the survival and productivity of different species, starvation, predation, environmental stress, other types of things that have some sort of a bioenergetic underpinning to it. And so we can deal directly with what's the effect of quantity or quality of food. And I do want to talk a bit at the end of this this talk about access to food. You heard about this earlier. There's that component that's vulnerable of a prey base. There's that component that may not be vulnerable. And I think that's a really underappreciated part of food web work so far. And so dealing with that, I think, is a real point of emphasis. So things like how thermal regime and environmental stressors might affect the distribution of organisms and how they perform in different habitats is an important element that we can use bioenergetics for. And then, of course, are these trophic interactions important in regulating populations? Again, whether it's direct or indirect effects on consumption, demand versus the supply of food through time, predation, mortality, competition, the dynamics of food supply overall. And so the nice thing about energetics models is they help us link behavioral and physiological characteristics of organisms and individuals that we can then scale up in time to higher levels of, the population level. It has a whole size structure and age structure to it, and then community level interactions when we start including other species there. And so some of these, kind of the core of these energetic interactions are some of these temperature and weight dependent functions there, among others, but those actually capture a lot of the important dynamics that we really want to look at. And we can consolidate those into a map of how the combined effects of food supply and temperature affect growth of an organism there. And this is just a juvenile Chinook salmon where the top curve, these characteristic dome-shaped curves with temperature help us understand some things, like there's no single optimal temperature for growth. It's a dynamic process that relates to what the food supply is in interaction with temperatures. And you'll notice as you go from feeding at an unlimited rate, that 100% Cmax down to 75 and 50% and below, that the optimal temperature for growth starts shifting to cooler temperatures. The other important element there too is that the thermal tolerance, or those temperatures beyond which an organism can't sustain positive growth, those also shift to cooler temperatures as well. And so this has ramifications in terms of what habitats can be used sustainably versus this push-pull of organisms needing to go somewhere instead. And so as an example for juvenile Chinook salmon, when they come into Puget Sound, there's this critical growth period that really influences their overall marine survival. The period over which they're in that pelagic marine environment really happens right around the plateau of these growth curves. So they're relatively insensitive to the effects of temperature across that range in comparison to the amount of food that's available to them. In contrast, if you go a little further north into the Strait of Georgia, the temperatures average about 2 degrees warmer. And so across the same range of temperatures, but just shifted 2 degrees warmer, those fish take a much greater temperature hit over the same range of food supply there. And so again, temperature can be really important in terms of where it falls within the thermal responses of an individual organism there. And then if we extend that to multiple organisms, we start getting some insights into whether there's segregation or concentration of predators, prey, and competitors there. Because if there are alternatives, say this predatory striped bass, if it has alternatives, it may try and stay other places. In order to interact with, say, juvenile salmon, they take a significant growth hit. Now, in some cases, like in river systems, there's no choice because you don't have that thermal heterogeneity through these migration corridors. But as you move into, say, earlier in their life in reservoirs where there's vertical thermal stratification, or out into the estuary where there's lateral heterogeneity, some of those interactions may vary because the overlap of predators and prey vary as a result. And the other piece of that too is just the impact of a striped bass. If these salmon are forced to move through and interact with striped bass when the temperatures are above 20 degrees C, those impacts are going to be much higher than, say, if they're moving through at 15 degrees C, where now that predatory impact is going to be maybe 20–25% of what it would be at those higher temperatures. And so when we put this together in a modeling framework where we're really looking at empirical inputs or expert opinion to try and feed these models, all these inputs in the yellow rectangle there are really the inputs that drive the model on a daily time step there. So thermal experience of the consumer, temporal change in diet composition, and ontogenetic changes as they grow into different feeding modes or different seasons. The growth of the consumer itself is a really important input that we're using to essentially ask the question, how much food did this consumer need to eat in order to satisfy the growth rate that we're observing for them? And then dynamics in the energy density of both the predator and the prey play a role in what the profitability of feeding on different prey resources are going to be. So given those inputs, that gives the investigator some flexibility in optimizing their study design. They can use either all empirically derived information, but now they have a sense of where do they want to invest most of their effort based on where some of the most uncertainties are related to either change in change in time or space or life stage, et cetera, there. We can also explore different scenarios by altering those inputs in thoughtful ways. And if we don't have that information, we can either invest in getting that information, or we can use expert opinion and literature values to do that. And so this model then cranks through that on a daily time step, and it pumps out daily estimates of consumption in terms of grams consumed of each prey that's in the diet. But we can then ramp that up to population level consumption rates, where we do that for each size class individually. So it still encapsulates all those changes through time and life stage. But now we're expanding that by the relative abundance of that consumer population. And then we sum all that together to come up with population level consumption through time and over the course of the year. And then if we know something about the biomass of the exploitable prey, we can start doing things like relating consumption in terms of a mortality rate, or as a percentage of the biomass of those prey or the production of those prey. Again, these are metrics that can be then fed into these more strategic level models like Ecopath, Ecosim, Ecospace, et cetera. And this is just an example of some of that. This is Lake Washington, and a couple of points I want to make here is that we've got the smaller fish on the left. These are less than 300 millimeter northern pike minnow. They're a predator in the lake. They're the most abundant predator versus the larger ones that are greater than 300 millimeters long. And of course, the larger fish are eating more biomass, but the diets change, and the diets change both among months within a size class, but the diets change considerably among the size classes. And most importantly, we were concerned about predation on salmon. Those are the reds and oranges at the top. And you'll note that only the larger pike minnow are eating the salmon. The other important thing here gets to prey access, and I don't have a great way of showing this other than to just talk through it, in that historically pike minnow are a cool water predator, and historically they track the warmest water in the lake available throughout the year. But in recent decades, the temperature has increased the temperature in the surface layer to the point where it's forced the pike minnow down into and below the thermocline. And by virtue of that, this circled area is now all new predation that historically didn't happen on juvenile salmon and longfin smelt and some of the other species there. So although the abundance of predators didn't change, that climate-induced shift in distribution increased overlap, increased interaction, and now we've got mortality rates that are upwards of 50% on both sockeye and Chinook salmon imposed by mostly pike minnow every year. We can also look at this in terms of the challenges confronting the spawning salmon in that they quit feeding once they enter fresh water. And so it's a race between the energy depletion that they're experiencing and their ability to get up and spawn successfully on their spawning grounds. And so this is just a simple look at this for sockeye salmon, where the colder the temperature, of course, the longer time frame they have to maintain their energy reserves. There's some important thresholds, though, that we want to be aware of. And one of those is that it's pretty important for these fish to reach the spawning grounds where they still retain about 60% of their original energy stores. And that's because they need an additional 15% to offload their gonadal material, as well as the activities of red building, defense, et cetera there. And the reason that's important is because there's been this kind of empirical threshold of at around 40 to 45% of the original energy content of these fish when they enter fresh water. Once they reach that 40 to 45% threshold, they're typically dying off at that stage. And so you can play all sorts of games with this in terms of, what temperatures these fish are exposed to, what swimming speeds they might need to maintain based on, either velocity barriers or flow shifts in the system overall, that can give us some insights into whether or not, these water operations are affecting the reproduction at that end of the spectrum. And so the bioenergetics tools themselves are really important in quantifying these linkages, driven by temperature, driven by the accessibility and quality of those prey. And it gives us a good way of identifying where these bottlenecks might be that could limit different populations, whether it's predation mortality, food supply, whether that food supply is exacerbated by competition, and how a thermal regime is structuring that overall. And what's nice about this is it often works in conjunction with routine sampling regimes for monitoring programs. Often they're getting size at age, diet, et cetera, or it can be modified in those programs to supply some of those really important inputs there. It's also great as a conceptual framework for integrating these data and identifying where those priorities are, i.e. those uncertainties, so we can quantify that and confront the management uncertainty that's going on. And the other key thing I think that hasn't really been used quite enough yet is that these bioenergetics models can simply be used as a module that's added to other models, like hydrodynamic models. We've done that to look at, say, bull trout or rainbow trout, how they're in reservoir, so 2D, through depth and through the longitudinal axis of a reservoir. How does their growth potential change through time and space? We can use foraging models to get more directly at how the predator is perceiving their prey, and I hopefully have a little bit of time to show some of that. And then you heard about spatially explicit models from Steve and Doran, I believe yesterday. This can be some of the underpinnings of that type of work. And so incorporating prey access, I think, is really important in terms of how prey encounter is really driving the ability for these fish to consume things. And so the visual environment, I think, is a really important thing to consider, because virtually all predators on salmon feed primarily through vision. A lot of fish, especially in pelagic zones, are feeding primarily by vision, no matter what trophic level they're at. And that visual environment is mostly affected by light and turbidity levels. And so understanding how that plays out through time and space, and including depth, is an important element. And one thing that we've seen is that you can have food webs that have similar assemblages, but they function entirely differently if the optical conditions differ there. And so there's something to be said there. And I just want to go through a couple of these things. This is a cartoon of in oligotrophic waters, you've got the search volume that a predator can transcribe through the water. As they go deeper, you've got this exponential decline in light that reduces that reaction distance, which is the red radius to that cylinder, and they go deeper and deeper. And the prey fish are not stupid; they've co-evolved, and so they adopt anti-predation measures like schooling or staying deep. And so the net effect of that is typically during those twilight periods of dusk and dawn. That's when peak predation happens. And in fact, this This bar graph off to the right shows that back in the mid 1980s, we did round the clock sampling of pelagic predators. Indeed found that twilight periods were when peak predation happened. And we can use these foraging models to try and quantify what those reaction distances are in time and space. What I want to say, though, is that artificial light at night has increased dramatically over recent decades, and it's created this essentially perpetual twilight. So now when we've gone 20 and 30 years past when we did that predation periodicity earlier, now we see predation happening through the night and happening deeper. And so when you think of this either in large pelagic systems or even in rivers and corridors where light penetrates to the bottom, that's an area I think that's rich for exploring for restoration efforts and other things. And because from our math, we know that the area of a circle is pi r squared. That reaction distance, as you change that reaction distance, that's a squared term. And so if we have something like a 50% reduction in light, or something that reduces the reaction distance by 50%, that original predation risk gets reduced down to 25% of its original. And so if we map out, say, surface light, and then this is Lake Washington again, surface light at night, and then look at how it attenuates through depths, et cetera, and apply it to various species, we can map out what that predation scape looks like through time and space and identify those hotspots. Use this as a way to identify both nearshore areas as well as offshore areas. The last thing I want to point out is that these, you'll notice that some of the hotter areas are out in the actual pelagic zone of the lake. And that's because one of the more important elements of artificial light at night is what's called sky glow. That's what's coming up and actually just reflecting off the molecules in the atmosphere. It's diffuse. It comes from up to 10 kilometers away. And it literally goes out across the entire pelagic zone. Those relationships I showed you earlier were from pelagic areas that were a couple kilometers offshore. And so this is real. We have the ability, by looking at how artificial light and other methods of visual ecology are affecting these fish, can actually have some real relevance in terms of how fish access their prey, either successfully or not. And if we think about this, we may be able to do system-wide restoration in terms of system-wide reductions in light, changes in turbidity, et cetera. And I'll stop there. Thanks.
Thanks, Dave. Take time for one question? Fred.
Thanks, Dave. Great talk. Question about reactive distance. How much do we know, I guess, from the literature and maybe just from your own work, do we know a lot about reactive distances for striped bass, largemouth bass, the typical predators that we have here in our system in the Delta? And with that, how interchangeable would you say are just the light aspect of it versus things that will interfere with light, say turbidity,
right,
like on your last slide?
That's a really great question. Yeah. So one thing, I used to be University of Washington, but I got recruited over to this USGS lab, and it was primarily because they had the experimental capability of dealing exactly with those questions and with these bioenergetics temperature control questions. In terms of light, you have a species-specific response to light. Even within the salmonids, we see some up to twofold differences in reaction distance as a function of light there. Turbidity, I think, is a much more system-wide, taxa-wide phenomenon in that backscattering really affects all species. Now that's buffered a little bit by some coastal marine species, for instance, have like some yellow filter in their cornea, which helps reduce some of that backscattering. But even with that, there's only so far you can take that. And it turns out that the predators, the piscivores, are much more sensitive to very subtle changes in turbidity than the planktivorous fish are. And if you think about it, the piscivores are operating at, their prey densities are on the order of 1 or a fraction of 1 per 1,000 cubic meters. Whereas a planktivore is in marine settings, it's like hundreds per cubic meter, or in freshwater, 1 to 5 to 10 to 15 per liter. And so you can really constrain the optical environment without impinging on the invertebrate feeding fish, whereas the piscivores are extremely sensitive to that. And so there's an area where that asymmetry can really be used to advantage in terms of protecting those intermediate trophic levels there.
Okay, thanks again, Dave. And I want to thank all of our speakers for really giving us some greater insight. Bob, did you have? Okay, great. And we'll break for coffee.
Billy, are you ready? We're ready.
All right. Okay, if everyone could take their seats, we're ready to go here for our lightning talks. We have 7 speakers. They have 5 minutes each to tell us about new tools and methods that are out there for assessing food webs. We're going to hold questions until the end, so we'll move right along through our seven presenters, and I'm sure there'll be plenty of questions. If we don't get to everything here in this hour, we have another breakout group right after lunch, which is going to be focusing on new tools and methods. So I'm sure we'll circle around again and come back to many of these. So I'm going to get this moving by asking Melinda to give us a start here. You can, well, on this, red light on and speak into the microphone.
Okay. All right. Hi, I'm Melinda Bearwall, and I'm from the Department of Water Resources. And today I'm going to be talking to you about some key considerations when deciding if genetics is the right tool for you in predation studies. Oh, forgot. Okay, so it is challenging to meaningfully detect and quantify predation since the majority of predation obviously happens underwater in our system. And so we often use either gut contents or fecal analysis for the work. That itself poses its own challenges because prey can digest quite quickly. Even when prey are detected, it can be difficult to be able to identify them down to the species level. Therefore, a host of different tools have been developed, and each has their own taxonomic biases dependent on predator and prey size, digestion rates, hard versus soft prey items, and other factors. One of the primary tools that is used is visual inspection, which can often be done with microscopy or with the naked eye. And this involves looking for recent total prey items or identifiable hard parts. Traditional microscopy can be very time consuming and has associated costs with it. It requires specialized skills in that type of taxonomic identification and can have issues with accuracy, getting down to the species level, as well as sensitivity issues. Contrastingly, genetic tools can have a reduced time because samples are processed in mass, and we are able to use very high throughput equipment to be able to analyze dozens, if not even hundreds of samples almost simultaneously, especially with the right equipment, therefore reducing both time and cost. Accuracy can be increased because you can get to the species level identification. And what we do basically is use the DNA of the prey found in the gut contents or feces for that analysis. Over there on the lower right, that is what we scientifically call digestive goo. And it is something that usually you would not be able to use for actual visual identification, but we can use it for genetics. And then we did a captive feeding study where we fed larval delta smelt to silversides. And what we found was that we could genetically detect the prey up to 9 hours with very high rates of reliability post-consumption. And even a day and a half after they were consumed, we could still detect it in some individuals. This is in contrast to literature, which shows that in microscopy studies, you can usually not detect it 1 hour, past 1 hour post-consumption. So quite a difference. We, in our most recent study, looked more holistically at predation patterns by using 6 different predator species taken from throughout the Delta, different times of year, different places, and looked for the presence of 10 different prey species in their gut contents. We were looking at different things such as the predation patterns on native species, as well as spatial and temporal trends to those patterns, and any habitat conditions associated with predation. Particularly, we were interested on native species. I don't have time to go into all the results today, but I will say that we found an interesting thing with striped bass, where they actually consumed native prey at higher proportions than what we anticipated. 60% of the diets were native prey. The reasons for this are largely unknown, but it could be things in terms of the prey recruitment patterns or prey preferences. I encourage you to review our paper for more information for that. Where's the laptop that it was? Oh, it's over. Okay. I'm like, it was right there. Okay. So when trying to decide if genetics is the right approach for you, there's a couple things to consider. And that includes that you'll want to collaborate with someone with genetic expertise. There can be a large amount of development time. It can take a while. It all depends on the genetic resources that are already present and available for your target species. There's also, it can be issues with contamination. This particular method is so darn sensitive that you need to take precautions at all stages of the process to avoid getting false positives. And last but not least, this type of data usually relies on presence absence information. So it tells you, is a prey present or not? And we don't typically do quantitative output in terms of saying this many prey were in the guts. And we also don't say it was an egg that was consumed versus a larvae or some other life stage. With that said, we have been able to make significant advances in this field for a genetic standpoint of quantifying prey consumption. And a particular area that is quite interesting is DNA mixture analysis. And this is largely done in crime scene investigation work, where you can take a mixture of DNA pool and figure out how many individuals were comprised in that pool of DNA. And so down at the bottom, what you can see is a study that used 3 Chinook smolt individuals that were fed to individual channel catfish or largemouth bass, and then the investigators tried to genetically estimate that number back. And although the results here in this graph show that it was not perfect and more development is needed, this is not bad either. And so I do believe that this is a very promising avenue to look for, not just to be able to use genetics for sensitive and accurate prey detection, but also perhaps in the future prey abundance estimates as well. So last but not least, I'd like to, Brian Schreier and I, my co-lead on this, would like to thank a variety of different people that have worked with us on predation studies over the years, as well as our funders and you for your time. Thanks.
Great, great. Thanks, Melinda. Sounds like a wonderful technique. So Matt, you're up next.
Does this work? I've never done this sitting down. Okay, we good? I'm going to be talking today about eyes and use of fish eye lenses as a tool to recreate diets across ontogeny. So Ryan Bellmore mentioned it earlier, but the lens, the eye lens of a fish, is highly proteinaceous, sequentially deposited, and metabolically inert, meaning it's an excellent archival tissue for keeping track of anything that can be recorded in protein throughout a fish's life. There's some really cool biology comparing like fish eyes with other vertebrate eyes. I would love to talk about it, but I have 5 minutes. Like, as I said, lenses are sequentially deposited, meaning they have multiple layers. Think like an onion, that's like a oversimplification, but these layers can be mechanically separated by hand or cross-sectioned and evaluated that way. And you can get a lot of valuable information about an organism's habitat use, lifetime diet, or contaminant exposure through these things. Examples. So just looking at habitat use and lifetime diet, often this is done with light isotopes, think carbon, nitrogen, sulfur, but we can use other tools if needed. For example, we used eye lenses in a large native minnow hitch in the Clear Lake here in California to identify ontogenic changes in habitat use. So throughout their life, they start out in nearshore littoral habitats, and then they went ahead and shifted to offshore pelagic feeding, validating answering a lot of uncertainty about the life history of these species that we couldn't have got other ways, or that would have been extremely intensive to address in other ways. We can look at other ways to address habitat use. So here in the Sacramento-San Joaquin Delta, Miranda Bell Tilcock and others have used sulfur as a tracer to mark floodplain rearing in salmon. And through this, you can see different cohorts of fish which have reared on floodplains versus which have not. This can be used to answer all sorts of questions about growth, survival, etc. But this is a tracer that would get you information you wouldn't be able to get in a different sort of way. This has even been used to reconstruct lifetime diet for some of these same salmonids. So Chinook salmon leave the system, they go out to the ocean, they come back, and we don't know what they did in between. So Rachel Johnson and others are using isotopic composition of eye lenses to recreate their marine diet and evaluate some of the potential for thiamine deficiency and other marine-based issues to influence return and survival. Additional questions on that, I defer to her and her colleagues. Another thing that you can use eye lenses for, because they're proteinaceous, they're excellent at recording and binding certain contaminants. So we've done some work, again, here in the Delta, looking at selenium exposure in these fishes. So there's been a lot of work using otoliths to document high levels of selenium, both environmental and maternal, in Sacramento splittail. And then we, using these same fish used for these otolith studies, are able to document that eye lenses incorporate selenium the same way, giving you the potential to evaluate dietary sources of selenium or potentially a cheaper or simpler method to evaluate this toxicity. Same sort of thing goes for mercury. So moving back to Clear Lake, mercury is one of those things that's really difficult. The ultimate source of mercury is really difficult to resolve sometimes. Sometimes it bioaccumulates. Also, sometimes it can actually be excreted or removed from tissues. But using eye lenses, you can actually trace ontogenetic mercury accumulation through time. So this is just comparing mercury accumulation in sunfishes in Clear Lake, which they shouldn't have much, and they don't. But then largemouth bass, a highly piscivorous fish, which will have a very clear uptake in mercury concentration, typically about when they start getting really piscivorous, 150 to 200 millimeters. There's a whole lot of nuance to this. I would love to talk about it. This is not the exact time to do it, but please hit me up if you want. There are a lot of people that work on this stuff. Happy to talk about it at any point. Yeah.
Great, thanks, Matt. Amazing technique. So our next presenter is Levi Lewis, and he's going to be online, virtual. So he's up there.
I should be sharing. Can you hear me?
Okay. Very good.
Are you able to hear me?
Yep. You're
Wonderful.
all set. Go ahead.
All right. Thank you. Thanks for bearing with me while I fight this virus. Today, I'm going to talk about how we might use otoliths to inform food webs. Let's see. There we go. So I'm Levi Lewis, PI of the Otolith Geochemistry Fish Ecology Laboratory at UC Davis. We combine field surveys with laboratory analyses of otoliths, both to look at age and growth and movement patterns using otolith chemistry of a variety of different species of fishes, from smelts to salmon. If you want to kind of dive into the details of otoliths a little bit more, I strongly recommend this paper by Patrick Reis-Santos. He goes through 7 different applications of otoliths, and number 5 is about food webs and species interactions. So there's a lot of really valuable information in there as well. Okay, so otoliths are ear stones or small bones inside the heads of fishes. You can see 2 in this longfin smelt larva. There are 3 different pairs per fish. The shapes vary among species. They're used for hearing, orientation, sensing movements. They're comprised primarily of calcium carbonate with a mixture of organics and various other elements. There are several different processing steps that are required to get any kind of information out of them. But once we kind of specialize and refine these, there are lots of different kinds of data that we can get from otoliths that have direct application to food webs. So informing predation, one thing that Melinda pointed out is that sometimes you get these weird kind of gooey gut contents that are hard to identify, or you get highly degraded fish, you don't know what species they are. But what's really nice is you can pull these otoliths out, which are pretty resilient to digestion, and fishes have very specific shapes with respect to the otoliths, even within a genus, even across species within a genus. And so you can use the otoliths as another tool like genetics to identify what species of fish were in those gut contents, which is really nice. You can also extract the organic fraction from otoliths and create isotope biplots or triplots like Matt was just talking about. There's very little organic material in odorless, and so eye lenses are probably a better tool for that, but odorless is provide another option for those kinds of analyses as well. And you'll see in the literature that folks are using odorless to look at metabolic rates and temperature history using inorganic fractions of 13 carbon and 18 oxygen. But I would caution that most of those studies are in very stable environments like deep ocean or open ocean. In estuaries, the baseline is so variable, it makes that really difficult. So just keep that in mind if you do see those kinds of papers and are thinking about applying that to the estuary. Okay, with respect to production, odorless provide several different ways to look at growth and mortality. We can look at age structure, size at age, mortality. We can construct von Bertalanffy growth curves for fish, which gives us instantaneous growth rates as a function of age or size. We can also create individual growth chronologies for each individual fish. So for example, odorless size scales linearly with fish length. And that allows us to convert daily growth rings in prey, like delta smelt, into daily growth rates in terms of millimeters of fish per day. And similarly for predators or larger bodied fishes, we can also use annual bands to construct annual growth rates and annual ages as we age kind of from the core to the edge, which represents the entire life history of that organism. And that allows us to provide all these different types of metrics that are taxon specific empirical values of mortality, growth, and all of this can be scaled up to biomass production, all of which can be informative for some of these more complex ecosystem and food web models. And then the last is provenance, and this is where we're using geochemistry to kind of trace where fish are coming from and to where they're going. So for example, in the Sierra Nevada, I don't know if I'm my laser here, in the Sierra Nevada over here, these rivers have distinct strontium isotope signatures, many of them do. And here on the left, you see the strontium isoscape of those different river systems. And we can use that isoscape to place fish into their natal streams. And these could be juveniles that are out migrating, or they could be adults that are returning to spawn. Thus, that allows us to understand where fish are coming from as they're moving to the ocean, and also whether or not fish are straying or are spawning in habitats that didn't come from as they start to return to their rivers to spawn. And this is particularly valuable for things like salmon that make these large migratory runs into all these different rivers. Similarly, we can use strontium isotopes to also trace salinity. So in the estuary down in this region, we have a nice salinity gradient from the delta to the ocean, and that gradient is strongly correlated with a gradient in strontium isotope ratios of 87-86. I won't go into details about why, but we can use that to trace the migratory history of fishes that are moving across that salinity gradient. So species like sturgeon, smelt, and salmon are all moving across that gradient, and we can retrace their life histories to inform us a little bit more about what those individuals are doing and how they're using habitats. So a couple examples with respect to provenance. So we recently published, well, I guess not recent anymore, it's a few years ago, but in 2019, we published a paper on delta smelt confirming our suspicions that there were different life history contingents. There were freshwater residents in the red that are staying in freshwater. There are brackish water residents that are basically being born or moving very quickly to high salinity water and staying there. And then there were migrants, which were the dominant life history that were born in freshwater and then moved to brackish water habitats as adults. So the life history of delta smelt, previously described as semi-anadromous, is a little bit more complex. We have several different several different contingent types that are doing different things, feeding in different places, and possibly interacting with different food webs or different parts of the food web. We recently published another paper on sturgeon, kind of doing the same type of analysis, and we found, again, different contingents or clusters of sturgeon that are doing different things, from the extreme of many fish staying in fresh water for over 10 years, and to others that are moving very quickly out to brackish or higher salinity habitats. Again, those individuals are doing different things, feeding on different food webs, and that likely needs to be accounted for in some food web models. And we even ventured away from fish and started looking at an endangered species of bird. This is a least tern, where we collected fish, where we, Point Blue, collected fish that were dropped at the rookery, and we analyzed the chemistry in the otoliths, and we can identify which hatchery those fish came from, the prey. We can identify whether they're wild or hatchery, and we could also identify which systems they came from. So we can identify the fact that predators may be preying on different contingents as well, and taking that into account can be really important for food web models. So contingents are foraging in different places, likely in different taxa, and transferring nutrients between otherwise distinct food webs. And otoliths allow us to dive into that. So in summary, otoliths allow us to identify who's eating whom, helps us understand biomass and energy flow, and then helps us think about these species interactions that may be a lot more complex, especially for fishes that are migratory or exhibit complex life history diversity. And I'm happy to follow up Thanks again for letting me present remotely, and I'm happy to follow up with anybody who has additional questions anytime. Feel free to send me an email.
Great, thanks, Levi. Like a really useful method. Lance, you're up next.
All right, can everyone hear me okay? So my name is Lance Takata. I'm a researcher from UC Santa Cruz and an affiliate with NOAA Fisheries. I'm going to be talking about predation event recorders right here. A PER, also known as a PER, an effective and adaptable platform for studying predation across a variety of aquatic environments. So what is a PER? So like I said, this is a PER. It essentially has two main components. It has a live tethered prey that you use as bait, and that's attached to a timer activated by a magnetic reed switch. When you have a predation event, the prey is pulled as well as the timer, as the magnet. This activates the timer. That way a researcher knows if a predation event has occurred and also when it occurs, when it did occur. So what makes a PER so awesome is that they're highly variable in the form that they can take. For example, this one is a free floating PER. You could have a lot of additional attachments to it as well. You could have environmental data loggers. This is for temperature. You could also have GPS tracking on it to actually track the location of your PER. And then you can also have video cameras if you want to record the predation event itself. Over the years, we've developed a lot of different forms. This is an anchored version if you wanted to sample a single location continuously. Here is a castable version. We call it a micro PER or an M-PER. It's really good if you can't reach a sampling location by boat or shore. You could actually use a mid-weight rod to cast to that location. You can even have GPS on these as well. Here is a shore-mounted version of a PER. Again, all the same components. It's just attached to a pole this time, and it's a fixed point on shore that we've developed. Where can PERs be used effectively? Pretty much anywhere predation occurs. We've had really good success in the Delta and Lower Sacramento River, but you could also use it. You could also use it in lakes, estuaries, marine environments as well. I believe the poster over here from UC Davis has it used in a wetland. Again, you can see all the previous forms that we've developed already and the ways that we've deployed them, so very adaptable to different environments. But most importantly is the data that you're collecting. You're not getting a true predation amount, but a relative predation amount. So really good if you have control and experimental groups or sites or treatments. And then additionally, you could use a Cox proportional hazard analysis to analyze the influence of environmental covariates on your predation risk. So for example, we often compare it to temperature, like on the graph on the left, or predation risk relative to, in this case, diversion distance, so distance to a diversion. And you can see how predation risk has gone down the further away you get from your structure. Additionally, I mentioned GPS tracking on your PER, which is really convenient if you want to actually, you can use it to locate the actual specific point of predation, and you can do a lot of cool mapping in GIS for that. So on the bottom left, you could see a heat map of predation in a study site. And then on the bottom right, you could actually see the tracks that a free-floating PER, that multiple free-floating PERs have taken, and the red dots indicate where predation occurred along those tracks. You can compare that to the middle picture. That is the same study site, but that's a habitat map that we did concurrently. And so you can use the two to kind of inform your decisions on where predation is occurring relative to habitat features in your study site. Additionally, you can, if you have video cameras attached to your PERs, you can do a video analysis to actually identify your predator species. This could be really important if, especially in the context of this workshop regarding food webs, is your predator a nearshore resident species like a largemouth bass, or is it a more free-swimming, deeper water species like a striped bass? Lots of previous literature already published on using PERs, so it's highly tested. Previous studies have included artificial light at night, predator removals related to predation, also predation around water diversion structures in the Delta. Most recently, we did an invasive submerged aquatic vegetation removal and looked at predation, how that changes. And then finally, I just want to, a really quick clip. This is from a pole PER in the Delta. And so we use juvenile Chinook salmon, but you could see the striped bass going after it. But I just want to reiterate that PERS are really effective at measuring predation risk. And they're really effective because they're highly adaptable to a variety of environments that you want to use them in.
Thank you. Very sophisticated. Okay, Vamsi.
Thank you all. My name is Vamsi Krishna Sridharan. I'm a water resources manager at Tetra Tech. Today I'm going to be talking about work that I did when I was with the University of California, Santa Cruz and the National Marine Fisheries Service. We're shifting gears from laboratory-based and field-based methods to a numerical tool. And I'd like to say in jest that I traveled 3,000 miles to talk about a model that's going to simulate a 100-kilometer journey. So the idea here in terms of this workshop is, can a salmon migration model be coupled with other trophic levels to figure out if you can do better in that migration model? So one of the issues with these migration models is mortality is typically simulated using some sort of mean field model. In this case, the Anderson-Gurari XT predator-prey interaction model is used, which essentially is an exponential decay function that depends on space and time in terms of distance that these simulated fish travel and time they take to travel between expected predator encounters. So I just want to hypothesize whether we can do better than that. So most of this work is going to be prognostic about things we could do with the model. The model is published and is available. The GitHub repo is indicated at the bottom here. It's being used to make biological opinion decisions on water management in California. It's a numerical test bed where you can code up behaviors as a function of the environment that the fish migrate in this case, salmonids, and use that to simulate how these fish move through the system. In this case, the Sacramento-San Joaquin Delta. The underlying model is a hydrodynamic model on which the behavior model sits. So it's an individual-based model. So there's many, many definitions you can give to a model of this nature. The migration model has 5 major components: swimming behavior, holding patterns as a function of tides and day and night cycles, orientation relative to the ocean, predation, and routing. This is just a slide showing how the model is performing when you compare it with out-of-sample validation. So the figures on the left side of the on the left side of the slide show you the routing performance as a function of tide phase. The figure on the right shows you survival through the delta from Sacramento all the way down to Chips Island as a function of mean Sacramento River flow. And you see that the model typically captures observed data and mark recapture methods based survival really well. So the idea here is that I have a video here. Can I come up there and play this? I think do it. Okay, thank you. So the idea here is that we can potentially simulate a variety of behaviors. So what you're seeing here is the convex hull of 2 different types of particles that we are simulating. The one in pink represents movement of juvenile salmonids. The one in blue represents movements of passive particles. And they're both particles that are released from the San Joaquin River, and that's superimposed on a flow field that's switching colors from reds to blue, indicating the phase of the tide either going out or coming back in. And what you see here is that with this kind of behavior spectrum, you can essentially simulate different species of fish. Where do we go with that? Recent work by some erstwhile colleagues of mine and probably continuing work in Steve's lab is indicating that you can build really sophisticated models of predator-prey interactions that take into account the sensory perceptions of predators and prey and have some sort of a proportional control between these predation events. Similarly, salmonids that are migrating through the system and that have to eat their prey, nectons, also are shown to be exercising this kind of proportional control. So from this sort of model, we can establish a probability of predation events being successful, and thereby, based on your local environmental conditions, construct more granular predation risk that can be put into a model of this nature. And one of the nice things about these predation events is that they are so rare in comparison to the amount of a species that's being eaten, so that we can simulate these different species of fish as though the other species weren't there. So let's just take an example of 3 trophic levels: necton, salmon, and its predators, say largemouth bass or striped bass. So I could simulate the movements of necton in some simplistic manner, probably a diel raising and lowering pattern within the water column. I have a calibrated model of the salmon. I can simulate its movement through the system. With striped bass and largemouth bass, some of the work that Cyril Michel has done, and some of the work that talked about with the predation event recorder indicates that they have some specific areas in the water column where they like to populate and how they move about in the water column. So those can be potentially simulated as well. So once we have those simulations, we'll have fields of these different trophic levels within the delta. And then on top of that, you can superimpose these more complex predation models that give you a very high fidelity and high resolution window into where different probabilities of mortality are happening. So what I'm showing here on the right is a simulation of the base EPTM, which is the version of the model we have right now that doesn't have all this fancy predation-related mechanics, but it indicates a path forward that we could potentially use this kind of trophic level interaction from these fine-scale studies to enhance this model and truly make it an enhanced particle tracking model. There are other applications for this kind of model. Melinda talked about the role of eDNA in identifying diet content and assemblages. Melinda and I have spoken about using hydrodynamically inspired particle tracking models to simulate the movement of eDNA in the environment. So that can be potentially coupled with the particle tracking model so that we know a source that is potentially shedding the eDNA as it moves through the system. And we also have a simpler particle tracking model that represents the movement of eDNA through the system, which would essentially be a settling model. So with that, we could potentially simulate or at least try to bound the box of uncertainty on where this eDNA came from. Just before this panel, Kenny, Steve, and I were having a conversation about viruses. And so one of the things in the Sacramento-San Joaquin Delta is the prevalence of this worm called C. shastra, which is a parasitic organism that sometimes infects the salmon. And so again, an application of this kind of model could be, I could potentially simulate virus particles as particle particles moving through the water. I could simulate C. shastra with some simple behaviors, and then I could simulate the salmon model. And these 3 potential trophic levels in this disease context could be coupled by an exposure model. So there are many applications for a model of this nature. It's just a question of identifying where you want to expend your energies. And with that, I would like to thank my collaborators and our funders, the California Department of Fish and Wildlife, Bureau of Reclamation, and some of our collaborators include the Department of Water Resources as well. The model is in the public domain. if you're interested in working with us on this model, please do shoot me an email, and I'd be happy to discuss potential collaboration opportunities. Thank you.
Great, thanks, Mansi. Hey, is it Shruti? Very good, you're up.
Hi, so I'm Shruti Khanna from the California Department of Fish and Wildlife and the IEP synthesis team. And I'm going to talk about remotely sensing submerged and floating aquatic vegetation in the Delta. I know it's not as directly food web related, it's more, it influences habitat, and habitat influences the food web. So it is important to know about, and there are, oh, there. Thank you. Okay. Yeah. So there are a lot of existing data in the Delta that have been used to map macrophytes and floating vegetation in the Delta. These are just of the few that actually have been used for that purpose. There are more sources of data that could potentially be used. Some of the oldest data that we have go back to 2004. And this is hyperspectral spectroscopy data that have been used since by the C-STARS lab at UC Davis, and we've used it. And in some form or the other, they have been available, these data have been available all the way to the present year and will be captured again in 2024. Right? Yeah. So it is one of the oldest data sets around, but it's not complete. There is a data gap in between, which we have tried to fill with Worldview 2 imagery, which is satellite imagery. And most of this data is somewhere between 2 to 5 meter pixels. So it's pretty good resolution. And it is all now published online for anybody to use. Other than this, there is always Landsat, which goes back to, potentially 1978. There have been a few tests done with Landsat data, but we don't have a working mechanism as of now to map macrophytes with Landsat. It can be done. And then some of the upcoming tools are UAV for sure, which DWR is really developing their capacity to do UAV related mapping. Or drone imagery. But there are trade-offs between all of these kind of data, which can broadly be categorized into spatial, spectral, and temporal characteristics of all of these remotely sensed data. So for example, satellite imagery is usually the coarsest spatial resolution. The airborne hyperspectral imagery is finer than that, and UAV is very fine spatial imagery. However, that comes at a cost because UAV imagery cannot really be used to map the entire delta, for example. And there is a paper by Bolch et al. That actually goes into a comparison of hyperspectral versus UAV imagery if you did decide to map the entire delta using UAV, which is kind of impractical. So there are use for all of these kinds of data, and some of these spectroscopy data, for example, has the finest spectral resolution. The satellite data, less than that. The UAV data has the least spectral resolution. Apart from that, there is also a question of temporal continuity. So the satellite data, for example, so this is an example of some of the hyperspectral data that exists in the Delta. It's taken mostly once a year because the cost of getting this imagery is about $200K for the entire Delta for a single year. And then if you use satellite data, what you have is at a coarser spatial resolution and spectral resolution, but you have much better temporal resolution. The imagery is collected every 5-6 days, or for Landsat every 16 days. And you are able to actually, so this for example, is showing on the top of that plot, life history of Agaria. right? So you have all of these different stages of growth and senescence that you can actually capture using satellite imagery, which wouldn't be feasible using other kinds of data. I'm going to come to this last. This should have been my last slide, but I jumped the gun. So just showing a couple of examples because this is supposed to be a 5-minute talk. So one of the projects that was actually funded by the council was to use Sentinel-2 multispectral satellite imagery to map macrophytes in the Delta. And as part of that, as I was talking about, you get an idea of the phenology of these different classes. And Ade et al., the PhD student who worked on this project, was able to show that water primrose, a floating invasive in the Delta, actually has the widest growth period of any of the other major forms or communities in the Delta, which gives it a competitive advantage. So we can start to look at invasion strategies of these different species using these data. Another major project that is up and coming is a NASA project that was recently funded to actually operationalize Sentinel-2 produced macrophyte maps in the Delta to the extent that it could technically happen in perpetuity. So we want to make it so that it's automated and the data are published regularly. So this becomes kind of a monitoring tool that with very little maintenance could keep going into the future. So the NASA project has been funded for 4 years, and part of that project is to build species occupancy models of both floating and submerged aquatic vegetation. Here is some preliminary work on that I'm showcasing. What you see here is a relationship between where you will find submerged aquatic vegetation, for example, in the depth and velocity space. So what you have in blue is a probability, or purple rather, the probability of submerged vegetation is 100%, and what you see in the red and kind of moving into the gray is a probability of 0. And by doing this kind of, supposed to move ahead. There we go. By doing this kind of species occupancy modeling, we can start to see, even in the Delta, in different kinds of habitats, what are the different occupancy characteristics of these species. So, lowest probability of finding them in channels, but higher probability in slow, shallow regions versus in flooded islands. And then the importance of depth and velocity is different across those habitats. So these are the kind of things that you can start doing with these data. And now I'm going to go back to that slide. So overall, all of these different kinds of data that we have in the Delta have been used to, starting out with status and trends, but look at invasion strategies of these different invasive species, to look at their environmental impact, to start to evaluate management actions and their effects on these invasive species. And then going into the species habitat modeling, so we can predict what happens with climate change, and also to look at restoration success. Thank you.
Great, thank you, Trudy. And finally, but very importantly, Rosemary.
All right, thank you very much. So I'm Rosemary Hartman from Department of Water Resources and the Interagency Ecological Program Synthesis Team, and I'm here to talk about data because I like to describe my job as knowing where all the data are and putting them together in interesting ways. And we've made a ton of progress that has made food web science a lot easier. First of all, we have a lot of data, and the open science and open data movement has really picked up steam and made it so that all of these data, or almost all the data, are publicly accessible online. Any water-related data, which includes fisheries data, is required by law to be made available mostly on the California Natural Resources Agency's portal. All the interagency ecological program data from across the agencies is linked on our master data page. There's I should have checked, over 50 data sets linked there. USGS has a ton of data sets from the Delta that are made available on ScienceBase and NWIS and their other portals. The SACPass website is a clearinghouse for salmon related data. Cowfish has any data you could want from inland fisheries, and a lot of IEP data is published on the Environmental Data Initiative Data Repository, which is an archiving platform. More than I didn't reference here. But once you have those data, they don't always talk to each other. And even data on, say, fish or zooplankton might be collected with different methods. The formats will be different, and you have to get them talking to each other, which is not an easy task. But fortunately, the IEP synthesis team, frequently led by Sam Bashevkin in the back there, has put together a lot of these integrated data sets that takes, say, all the fish data that IEP collects, puts it in the same format, and makes it available online. So you can look across surveys, across time, across space at all of the Sacramento sucker catch, for example. Another integrated data set on water quality, one on aquatic vegetation, one on zooplankton, one coming soon on phytoplankton, and we have a couple more potentially in the works in the next few years. And one of the things that has made all those data sets so great is that they're compiled collaboratively and made available on GitHub. Both GitHub and the wave of increasing data literacy and data analysis literacy and coding skills in the scientific realm has really made the data science move ahead by leaps and bounds. Now I can collaborate with anyone using R or Python on GitHub to put together these complicated data sets and do some really cool analyses without needing proprietary software or having people who speak different languages. Once we have all the data together, we then need to analyze it. And there are just a slew of websites where you can visualize these data. Bay Delta Live has a bunch of different ways of looking at all these different data sets. CalFish Track lets you look at acoustic telemetry data in real time. The Delta Science program has put together several Shiny apps to let you not only visualize, but also query and download some of these integrated data sets that I mentioned. And the SACPass, which stands for Sacramento Passage Assessment of Salmon, something. Yeah, anyway, any salmon related data you want, and this is really used by managers to check on conditions in real time and make real decisions. CDFW also has visualizations of many of their long-term fish surveys. Once we have all those visualizations and data, the community has also been moving by leaps and bounds, and the increased number of online and virtual or hybrid meetings has increased our participation in collaborative groups. IEP has a number of what we call project work teams where we get together to talk about salmon or synthesis or zooplankton and put all of our data together and talk through things, integrate across disciplines, across agencies, across food web levels to bring out some of these patterns. I just want to quickly go over two use cases. First one, this recent paper by Rogers et al. Looking at top-down versus bottom-up drivers of food webs in the Delta. They used this was put together by the Delta Science Program and the National Center for Ecological Analysis and Synthesis. They got this interagency team together to look at food webs in the Delta, and they did a series of really cool, complicated models looking at cross-trophic levels and found a lot of really interesting patterns. This was a very academic exercise. It was not designed for a management decision, but it brought out a lot of neat relationships that could be used later. And then I wanted to show one where we did use it for a management decision. A lot of these integrated data sets went into a series of interrelated models to actually answer a management question. The Delta Coordination Group is a team of high-level agency managers who are in charge of deciding on the best course of action for the summer fall habitat action for Delta smelt. And they took hydrodynamic models, zooplankton models, and fish bioenergetics models and linked them together to look at the potential outcomes of several different management scenarios. These integrated data sets is also being used to look at the reconsultation on the long-term operations of the State Water Project, the ITP, and to analyze the effects of the emergency drought barrier and in environmental impact statements for several of these management actions. So they really are being leveraged for management decisions. We aren't quite there yet. There are some gaps. We don't have great diet data, or it hasn't all been integrated across sources. We could always use more of that. Large predatory fishes are undersampled in the estuary, but they're getting better with the enhanced large fishes survey that Jim Hobbs can tell you about. We also don't have great integrated data on predation rates, or really enough of those PERS data to use them in some of these complicated models. And then some things like birds, otters, crayfish, sponges, we just don't have the data on yet, but could in the future. And that's all I have.
Great, thanks, Rosemary. Making great strides forward. Okay, we have time for a few questions. Not too many, I see. Audience? Please.
Thanks, everyone. What a great panel. This was really exciting to see all this progress. And so thank you so much for sharing. I have two questions if I can be indulged. One is for Lance. I'm just dying to know what your recent work with the PER system showed with respect to SAV removal, if you were able to remove it, and then what happened to predation rates. And I'll just ask both my questions, and then you can talk. So the second one is for Shruti. Thank you for sharing that progress on occupancy likelihood of SAV given depth and velocity. It's really exciting. And I'm wondering if you can imagine layering onto that other parameters like temperature, salinity, turbidity, and being able to predict occupancy with multiple dimensions of water quality and conditions, and whether we're at a point perhaps where we could get to species specific likelihood of occupancy for SAV species.
Yeah, so to kind of give context, the recent project we did was in the Delta off of Bacon Island. We were doing a submerged aquatic vegetation removal for Ageria densa, and we had a sampling period beforehand, before the SAV removal, and then a sampling period after the removal. The SAV removal itself actually went very well in terms of how effective we were at removing it. Obviously, we were only removing we removed 3 100-meter stretches along the bank, which compared across the whole, we mechanically removed them too. So using like scuba divers and people in the water. So not a large area relative to the whole delta, but we were very effective in doing that. In terms of the results and what we were seeing, we were expecting an increase of, or this decrease of predation. We were expecting that by removing this dense habitat that a lot of nearshore predators like largemouth bass like to reside in and predate in, that it would reduce predation. In fact, it actually increased predation almost to 100% in certain areas, mostly due to striped bass though. By removing the SAV, it actually allows, I think, the striped bass to predate in these areas. You could see them often coming in from deeper waters and moving inshore, which was unexpected, but is still an interesting phenomenon that we saw.
So a corollary to that, did you measure turbidity after removal? Did it go up?
We did do some drone flights over the area as well to get chlorophyll and I think turbidity levels, but I don't know the data exactly on that yet.
Because I'd heard that maybe it was removal from riprap, was that right?
Yes.
Yeah, so most likely what the results might be slightly different if you had soft substrate, that the turbidity would actually rise, or an area where turbidity would show a big difference.
Correct. Yeah, because SAB is very influential in terms of the amount of turbidity. It tends to reduce it. But again, we were only doing 100-meter sections, so over a larger scale, you might see a different result.
So, yeah, so to answer your question exactly, yes. So we do actually that the preliminary data that we used were salinity, velocity, depth. Now we have turbidity, we have temperature from ecostress imagery, but also from Landsat thermal imagery that we are going to incorporate in there. And we are also going to develop the model maybe on 2 years of data and test it using a third year of data, because there are at least 3 simulated years, because the back end of this niche occupancy model is the untrim, the untrim hydrodynamic model that is giving us the velocity and and then we are also planning to incorporate depth using the DWR's hydroacoustic data, which is on a much finer scale than just the untrim model. So definitely a lot of changes being brought in, and hopefully, so we specifically want to bring in parameters that we expect to change from year to year to be able to do that kind of, climate predictions and those like predict a little into future using conditions that are likely to change. Because if we don't have those parameters in there, the usage of that model is kind of limited.
Very good. Great. How about one more question and then we'll break for lunch? Who wants to ask that last question? Andre.
Thanks, Andre Buchheister, Poly Humbolt. This is an easy one. I was just curious for Melinda and Matt. Could you speak to what the cost is to process samples using the genetics or the eye layers for diet purposes? Thanks.
So for genetics, it really depends on the tool that you're going to be using. We often, for the kind of high throughput that we were doing, we're using what's called a fluidime chip. So it's just a very tiny chip, and we can do up to 96 predator species with up to 96 prey. And so that's the most extreme. And I would say to do that would cost a few $1,000s of doing the actual resource. And then for the people, it would be their time. And so that would be another couple $1,000. So you can actually get a lot of data fairly inexpensively.
Regarding processing island samples. So a lot of the cost is tied to typical analysis for light stable isotopes, so carbon, nitrogen on a per sample basis. Depending on the lab, that's 10 to 30 bucks a sample. And depending on the size of the fish and the temporal resolution you're interested in, you could get anywhere from 3 to 30 individual increments from an individual fish. And there's a lot of validation. You'll need to do multiple eyes, like both eyes from the same individual for a subset. There's a whole bunch of stuff, but ballpark.
Good. All right, let's thank everyone for an excellent panel. That was really interesting. Okay, let's break for lunch and be back here at 1 o'clock. And for those of you who ordered lunch, I believe everything's on the back table.
All right, Diane, you can take it over here whenever you're.
So I know we're all looking forward to the report outs from the stimulating discussions we had at our breakout session. So we have 3 report outs from our 3 panels, the 3 breakout groups here, and then 1 from the online group. So we'll start off with Christine.
Okay. Is this on? Okay. All right. So we talked about a lot, so I hope my group will forgive me for focusing on the high level key points for all of the different questions. So when we discuss the types of models and tools that would be most effective, kind of the first response to this was, well, it depends. Like, what are you going to use those models and tools for? So there's a little bit of it seemed to me like there was a chicken and an egg situation, right? Like you have to understand what the question is before you start trying to decide what it is that you want to use to answer those particular questions and the types of models and the types of scales that you're going to be needing to look at. And so there was a lot of interest in moving beyond these linear relationships that are almost like knee-jerk reactions of, there's a fish that we're interested in, and so it's linear relationship. And so there was interest in, can we be more comprehensive? And so can we move into the types of models where we are representing trophic levels? There was a lot of interest in doing that. And my understanding from my group, I'm a modeler, so but my understanding is that there are currently projects in place to do this. And so hopefully we'll be seeing some of that work sooner rather than later. And so we didn't spend as much time on kind of the second question, but we did spend a lot of time talking about an ensemble model approach. There was this idea of maybe a hierarchical approach for how we do modeling the Delta would be more useful. And so this was referenced in some of the talks and conversations today, where it would be more useful to have these simple models where we're not investing as much resource in them, so that if we falsify those hypotheses, we can move on to something else. And so if we have a set of these small scale models that are across the Delta, we can use the understanding from those models to then feed into these larger, more complex models. So that idea of having not one model, and maybe making sure that when we talk about food web models, that we're not saying food web model and kind of implying that there is one model that we're trying to build towards, but rather accepting that we might have multiple models doing different things, having multiple different applications, and that's okay. And so the last thing that we talked about, we spent a lot of time talking about barriers, and I'll circle back around to the point that we made earlier, but the main one I think is worth repeating because it actually occurred yesterday with a different set of people, which is that we need a type of strategic plan. We need some kind of shared idea of why it's important to do food web modeling and how food web modeling, whatever approach we decide to use, addresses this strategic plan. And this is important for shaping priorities and making sure that there is staff and funding and resources available to do the monitoring that we need at the scales that we need to answer these large questions. Because that was one of the key barriers that we discussed first, is that many of these models are data hungry. They require a lot of people going out into the field. They require specialized skill sets in being trained in how to code and how to collect this data. And so there's a backlog of information, there's a backlog of synthesis. And so having that strategic plan and kind of making sure that people are aware of why it's important to continue these food models is important for making sure that we have the resources to address these questions. I think that is pretty much most of the high level stuff that we talked about, but if my group wants to speak up, feel free.
And we can hold off till the next presentation, Lillian.
Okay, so I will echo that our group, essentially for all four of the questions, the first answer that was always written down was, well, it depends on your questions, or well, it depends on what you're interested in. So with that caveat, we'll move on to the other things that we discussed that Christine didn't cover. So one thing that I thought was very interesting that came up in the first question was talking about not only, the motivation behind the models, but different types of models. So looking at the difference between strategic models and tactical models, and differentiating and making sure that when you're creating those models or developing those models, you're differentiating between those goals. Because they can be used for very different things. So for example, you're not going to use strategic models to create stock limits. And so making sure that you're differentiating with those models might be really important. There was a thought that maybe there is enough data to like Christine's group said, to use kind of these groups of models to better accurately represent the heterogeneity of the system. And it was suggested that, several nice models, so the intermediate complexity model types might be appropriate for doing that. Let's see. So also there was talk that in the Delta, a lot of the modeling is done for compliance reasons. And so again, just making sure that the objectives of the modeling is apparent when it's being developed. We also spent some time talking about modeling uncertainty. So actually that's more along the lines. So now I'll move to the second question. What level of complexity do food web models need to have? So again, we need to look at multiple scales, functionally different ecosystems, and integrate over multiple spaces and times. That kind of gets along to using a suite of models. But let's see. So we also discussed that there are kind of differences in short-term predictions versus equilibrium calculations. So looking at different time horizons and making sure that you're really differentiating between those is going to be really important. And again, these all have to be tied back to your management goals and your key questions. So we did have some ideas about it would be interesting to develop different scenarios that you test regularly. So you could either use the same model and test it at different time points, or you could use different models that you can kind of compare the results to learn about the accuracy of those models through that. And then moving on to the data that are needed to develop the identified models, again, with the caveat that it really depends on what you're going for and looking for. We talked about some of the new tools that were talked about today, like genetics, and also looking at having additional data and updated data for diet composition, as well as estimates of large predator abundances and kind of spatial location data would be really important. Also monitoring fish passage through open areas or kind of restricted areas like junctions. But we also acknowledge that there's a lot of data going on in the Delta that can be utilized, and we all heard about that today. So in this is a section where we had kind of started talking about some of the modeling uncertainty. So really being careful to understand when you're sticking within the capabilities of the model rather than going out to kind of the tails of the model where it may not be as accurate, but also really understanding the uncertainties of all the inputs and what the outputs of the model are. And then for barriers, we kind of ran out of time, but we generally thought that there is capacity in the basin to do some of these things, but really something like a collaboratory may increase capacity to understand all these things. So really having a consensus about the goals, the management questions, and kind of the priorities would be very, very helpful. In addition to just understanding the finances of how to do this and what's feasible.
Thanks, Lillian. So Kyra, do you want to go ahead?
You two ladies are a hard act to follow. It's not the best to go after you two, but I'll do my best. So in terms of the first question, what models and tools might be most effective, there were some sticky notes that said, things like hydrodynamic models and bioenergetic models, but I think kind of the broader discussion for this section was around, again, what kind of question are you asking? And there was definitely a consensus that the management questions that we're currently facing are maybe not, we use the word crisp enough. Maybe we need to focus a little bit more on honing in on those and exactly what we want to manage and what are our goals, and maybe figuring that out first before we get to that next step of, okay, how do we model something like that? So there was kind of a broader conversation about, the management questions that we're asking, and what we want to get out of that before we even get into the modeling bit. Let's see. And then in terms of the level of complexity that a Delta food web model might need, there was various sticky notes that had, spatial scales in terms of habitat type or region, temporal scales being either tidal or monthly or annual. So there was variation in that. But again, we had kind of a larger, more broad conversation about, you need both the simple models and the complex models. It's not necessarily one or the other. And there's different ways you can go about that in terms of starting on the simple side of the model and building up to the complex, or doing the reverse where you start with the complex model. And then depending on what your research question is, then kind of shaving off parts of the complex model until you get to the simple. So there's kind of various ways to get there, whether you're starting simple or starting complex. But there's definitely a clear need for both. So that was kind of the theme for that question. And then what data are needed? There was a lot of different things listed. There's, diet data, temperature, turbidity, gear, efficacy, biomass. There's a whole bunch of different things listed. But a couple key things that were talked about were that it's unclear how much diet data are out there. There's not a centralized database for that. That's maybe something that could be put together or standardized and would maybe relieve some of that lack of data we're maybe seeing in that region. And that we have a lot of prey data, but we don't have a lot of predator data. So kind of those are the key places that we might be missing. And let's see. So our barriers question, that was the section that filled up very quickly with sticky notes. It was very easy to get some answers on that. We didn't necessarily discuss it as much, but there was a lot of different things listed. And so some of those were resources and imagination. I kind of liked that one. Maybe widening our imagination on what's possible. Another one I liked said, need to convince people that this is not scary. So I think that was kind of a nice way to put it. Let's see. One said time. That's a barrier. It's running out on us. Let's see. Conceptual models of non-listed species. Inability to predict management actions, contaminants, data diversions, reluctance to change, lack of information on food quality. So there was a whole laundry list of barriers. So that one was pretty easy to get information on. So that's kind of just a very quick overview of that. So I'll stop there and we can go to our virtual group.
Thank you very much, Kyra. So we'll go to the virtual group now. And I think, Coco, if you just start talking, you'll show up. Go ahead.
Hi, can you hear me?
Yes.
Good. My name is Shoko Shinbrat. I work for the Delta Stewardship Council and happy to be joining you virtually. I am happy to see that a lot of the things that we discussed have actually already been captured. But we started our discussion in kind of a narrative format, thinking about, what is the appropriate level of complexity. One of the speakers advocated for, as simple as possible. And often, we talked about how systems are so heterogeneous, where complex models are good for, these high spatial and temporal resolutions, but we need to be careful if we don't have the data to build out the model. And similarly, I think this might have come up in every group. We need to make sure that the tools and data that we're collecting really align with what our questions are. So what are the questions that we're really trying to ask or to answer? And then we started talking about, making sure that whatever model is built is actually truly representing the system. So making sure that there's validation and calibration of the models. And it's funny, sometimes in these discussions, you end up getting more questions than you do answers. So many of my kind of key points here are actually framed as questions. And in, thinking about uncertainty, particularly, to what extent can these models be used for anticipating major changes to the system? This kind of dovetails with other efforts that the ISB is working on in decision-making under deep uncertainty. And kind of similarly, how can they capture and, reflect tipping points? In addition, we noted that, and I think other speakers have also noted that we're managing for a lot of listed species, but we might lose opportunities to manage for non-listed species. And then we took the opportunity to come up with some questions for the speakers, since we haven't had as much opportunity to chime in with questions. So some of the questions that came up were, to what degree have these models been useful in ways that go beyond what is expert knowledge? What are the management questions that need to be answered? This came up earlier. And to what extent can they be used for anticipating major changes? And then were they the best tool for the job? How does the design for the model matter, and maybe examples that have proven particularly useful? And then we closed out the discussion with major barriers, and just getting back to this question that has come up again and again of are we asking the right questions? All these models are designed to support decision making, but perhaps the questions are not necessarily what is needed. So this is a real barrier that we saw could be resolved through sustained engagement and sustained communication. And that's all I have.
Thanks very much. And right now the plan is to have a coffee break in 2 minutes. So, but we want to open it up for 1 or 2 questions before we do that, or some crosstalk across the different groups. Does anybody have a comment or a question they want to pose? I don't see any. Okay. Go ahead, Rosemary.
I just wanted to respond briefly to the question on how whether models give you more information than just expert knowledge would. And I think the value a lot of managers find in having a model is you can quantify it. If you're writing an environmental impact report or an incidental take permit saying that increasing the amount of pumping will be bad for salmon because Ted said so, doesn't hold a lot of weight, but saying we have this peer-reviewed, quantitative, complicated, multilinear Bayesian model that says increasing pumping will decrease salmon survival by 15%, that's something that holds a lot more weight with decision makers. Even if that 50% has huge error bars on it that often gets lost somewhere along the way, still having something you can quantify and then compare, okay, is increasing pumping gonna decrease salmon more, or is raising the water temperature or increasing harvest, like which of these has the potential to be worse? So, yeah.
Thanks for that comment. Steve, do you have a comment?
Yeah, just a little bit in response to that. I think one of the things models can do is really help you to see sort of counterintuitive results. And I think that we're commenting about the expert opinion, some experts can be wrong, and so can models. But I think that just looking at the example that we did on the Gulf of Mexico, where Kim and I were using a completely different approach, and I think common wisdom was when you cut nutrients by 40% and have all these improvements, you're going to see major changes in what's going on with the fish. And we both were coming up with 3, 4, 5%. So much so that I know I looked at my data many times to say where did I do wrong, and she came up with the same numbers independently using a different approach. So that's the kind of things that I think models can give you, is some of those bounds, some unanticipated results. You might have one species over here that is doing something that you never expected because of a secondary impact or something like that. So that's another value of these sort of approaches.
Stan?
This morning, Ryan made the comment that you learn so much when your models fail. And we rarely get up in these meetings and present the failure of our models. Has there been a retrospective of the models that have been used on the Delta and the failures and what was learned from those failures?
Yeah, this is Kim, the Mustard. I can provide one example where, you know, a ecospace model receives input from other models, and it's a food web model. So nutrients inputs has usually a strong effect. If you have more primary production, you have more secondary production. And I think it was useful already, regardless of what my output was for decision making, to noticing in my model that there was much more nutrients in the system than that was possible just because of the secondary production I was seeing, and going back to the models where that came from, where I was like, can I look at that data, right? It's just coupled and then looking at, well, there's no way there's that much nitrogen in the system if we make these changes. And it ended up changing that model to see like where is the mistake, and then we can run it again. So so I think sometimes by coupling these models, you can indeed, by crazy outputs, figure out like something along the way isn't going very well, and you can go back and figure out what it is and try and change it. So so yeah, I think definitely some instances where models failed have been useful in the process that way.
One more comment or question over here.
Just wanted to add about Vamsi Sridhar and Tetra Tech. I just wanted to add about the value of these models. So even when you have intuition or expert knowledge that something will happen, quantifying that will actually lead to proper management action rather than some sort of strategic idea about what you need to do. So I'll give you an example. So with the EPTM model, we were able to look at, it's not published yet, but we were able to look at different scenarios of pumping levels in the South Delta, and saw that, you know, at certain pumping levels, the entrainment envelope of fish is at, you know, a certain point in the Delta. At a different pumping level, it's at a higher point in the Delta. With a calibrated and validated model, when you have trust in what the model is doing, you can then take that to management action and say, maybe we need to put a barrier here so that in this particular place, so that fish do not get entrained into the pumps. Whereas with just expert opinion that if you increase the flow in the pumps, fish are going to get entrained, you really cannot say where you need to put that barrier. So that level of management precision, if the model is calibrated and validated, is definitely very beneficial to have.
Thanks for that comment. And this will be the last comment or question.
Yeah, Stan, thank you for your question about model failures. I don't know of like a synthesis of them, but it did get me thinking, and it seems like I can think of an old example that the entirety of striped bass population dynamics could be summed up in a linear regression. That was a model of the Bay Delta system at one time that did not hold up very well. And now I think maybe the newer one that I'm still waiting to hear about is there was a hypothesis for years and some very complicated models developed around the idea that the overbite clam had not wiped out all the chlorophyll production, but that a wastewater treatment plant in Sacramento was dumping out too much ammonium, and that was doing it. And so that led to treatment plant upgrades. To my knowledge, it hasn't led to phytoplankton blooms, but maybe there's a bunch of top secret information I haven't heard yet. So that might become one of the more famous examples of a model fail in the Bay Delta.
Well,
it-
as an environmental engineer, I'm going to say that whenever you upgrade a wastewater treatment plant, it's a good thing. So
Not arguing, not
Okay.
arguing that part.
So on that note, let's take our break.
How long is the break? Is it 15 minutes?
We'll reconvene at 2:40.
2:40, thank you.
Well, welcome back, everyone. And now that we have figured out exactly what the management questions are and exactly why food webs are important, and it's specifically what models we're going to use, it's you look so serious. I said that jokingly. It's time to look forward. And I'd like to introduce our next speaker, Peter Goodwin. Peter Goodwin is very well known in the Delta as a former Delta lead scientist, as inspirational and as an idea guy. So we've asked him to look into the future and give us some ideas on where we can go from here. Thanks, Peter.
Okay, well, thanks, Steve, for that welcome, and it's great to be here. Thanks for the invitation to being able to participate. It really has been quite a remarkable day and a half so far. I'm not sure how inspirational this will be. What is cut off on the bottom of the slide is that what I've tried to do here is to capture the ideas of many. And for those of you outside the Delta science system, this is an incredibly rich community, very, very well connected. And I would say very collegial most of the time. So what you're going to hear here is virtually nothing new. I've just tried to bring together some of those concepts as well as put it in the context of what's been going on with the federal government over the last 10 years. So I'm going to touch briefly, without undercutting the efforts of the Delta Science Program and Dr. Larson, just to give a very brief overview of these 3 decades of scientific community thinking behind, it's gone through many names, but an integrated modeling center. I'd also like to just mention the last 2 decades of investment preparing for the 4th paradigm, and I'll explain what that is in a moment. And then, as this is inspirational, certainly never in my career have I seen a window like we're facing now. And the opportunities that are out there are truly remarkable. And I'd just like to highlight a few of those at the end. And I'd like to conclude with this workshop perhaps being one of the founding grand challenges that could go into this type of concept. And I'd like to start with a tribute to Phil Eisenberg. For those of you who haven't worked in the system, Phil passed away last month. Remarkable individual, a true intellectual and politician, and he was tough on everyone. Great supporter of science, but he really asked some very, very tough questions, and they were usually the right questions. And having been on the tough end of some of those exchanges, there was one instance where he said, what was that, Lauren? But Peter, surely you know the Godkin lecture that C.P. Snow gave, and of course none of us had heard about that. And C.P. Snow is known for being a British novelist. He writes the spy books, but during the Second World War, he had been the aide to one of the 2 scientists that was informing Winston Churchill. And he gave this incredible lecture at Harvard in 1960. And for those of you interested, I could send it to you, because when Phil tells you to do something, you go and read it. But the point of this is he was talking about the intractable problem of bringing science into deciding really big decisions. The example here was, how do you defend the UK against aerial bombardment and the dispute that went on? And as he says, it's intractable. So very, very interesting, but it's essentially what we're facing. As we've heard, there's political and management decisions on one side, there's a science community on the other. Science is conducted on a very different time scale. the way we go about that makes it very, very difficult sometimes to have that best available information that Dr. Chakowski mentioned yesterday that's written into federal and state law. How do you organize that? And the first time I met Phil was in the Netherlands, and he was giving a keynote talk. And I think the title of his talk was something, "We're in Different Worlds." And this was something put together by the Ministry of Environment in the Netherlands, where they were trying to get the UN to give special designation to the estuaries around the world and the number of people that live around the estuaries. And Phil gave this remarkable talk, as I mentioned before, about this disparity between your management, policy, legal, and how you organize the science. And particularly in this system, for those of you who've been talking California in the US Congress, the story was just a few years ago. The reason that California can't get this figured out is because the stakes are so high. Literally every decision that our state agencies and regional federal directors are making, the cost during the 2011 to 2016 drought was measured in billions of dollars. That's a B. And so these huge decisions, and that's why no one can decide what to do. And so the Delta Science Program put together this very comprehensive document that described the complexity of the system, both from the social side, but also the science. Also, the role of the Delta Stewardship Council is quite remarkable, very, very supportive of science. They also have the responsibility of maintaining the independence of the Delta Science Program, which is why DSP is so highly respected across the board. And Patrick Johnson, a great supporter of science, described this same problem, and he referred to us as the monks of science, that we would sort of come down from on high and we would pontificate and then we would be gone. And how you pull those two together. And the image there, you could see how the ISB responded. You're in the next meeting. The fourth point here in this different worlds is that if we're going to solve these problems, we need to make really bold landscape scale decisions in the face of a lot of uncertainty. And if you think about it, if you're an agency director, if you're the head of a water utility and you're committing to these large scale efforts, we as scientists, if it doesn't work out, that's really good news. We need more study, we need more funding, and we'll figure it out. If you're the person making the decision, that could be the end of your career. So how do we organize science to empower decisions at that level? And of course, as we've heard, I won't spend much time on this, but often the scope of a science investigation that can be done, say, by a university with a particular single research project, or the agency mission is going to be restricted with the kinds of discussions that you've had over the last day and a half. And so the science program, with this really unique role, is putting together a strategy document. I'm not going to attempt to preempt from that, but the idea is to pull together the thinking of the last 3 decades, and many people in the room has been involved in those earlier iterations, to see how can we organize science, the decision-making process, to be more efficient and more effective. And the reason for putting all the logos on the bottom is just some of the efforts around the country where we've learned an awful lot how this can be done, whether it's through the technology side of Data One or the linking the social and the other science in a group, say, as succinct. And the important thing is every one of these logos, there's been very important contributions or leadership roles of researchers here in California. And so how can we organize that to take on community science? And community science is defined in many different ways. Here I picked one of my favorite definitions by Jay Lund, where he describes this as problems requiring developing and sustaining a confluence of scientific understanding among a very diverse community. And I think that really captures our challenge. And the problems which we take on, sometimes referred to wicked problems. This was in the report that DSP did, that these problems are too big and complex for traditional university consortia or a single agency to address alone. And as many of you know, and has come out in the discussion, some of these are approaching chaotic systems where you can have different outcomes given the same or very minor changes to initial and boundary conditions, and you can get a very, very different outcome. So how do you pull that together to make decisions? So stepping back now into some of the evolutions, and I apologize, I see a few faces here that have been involved in the evolution of cyber infrastructure through NSF and other federal agencies. But the thinking 20, 25 years ago, here I just picked one graphic to illustrate it. I apologize to Bill Mitchner, this cuts off his attribution on the bottom. But what was being noticed was that if you're a researcher, you have a highly productive lab, a very highly productive group. You publish something, but over time, there's a gradual diminishment of that expertise, particularly when people retire. This is a huge issue, of course, for us in the Bay. If you think of a number of the really iconic scientists that we've lost due to retirement just in the last 5 years, it's extensive. So how can you perpetuate that knowledge into the next generation and not be repeating some of the same ideas or the same mistakes? And here, I also just pulled out a few of the highlights from the pre-workshop community discussions. And what we heard was access to data. It's not just getting the data, but you need the metadata to understand what that data is and what sort of reliance you should place on it. And the other point I really liked that I thought came out very strongly, I called it team synthesis, but we make progress. But how do we track progress in our understanding over time, particularly in such a dynamic and complex system? And so a couple of other things just around this thing that's been driving the NSF cyber infrastructure. For those of you who've been following that, huge investments have been made in the connectivity between research universities linking to the supercomputer network that anyone in this room can now gain access to. And why would we want to do that? Well, E.O. Wilson, back in 1978, identified this problem, and I think we still suffer from that today. We have huge amounts of information, but we lack sometimes the time to develop the wisdom and knowledge. And so we're facing that. And then about 12 years ago, the mathematician that developed the algorithm, when you take your credit card and you put that into a cash machine anywhere in the world, and in a fraction of a second, you're pulling out cash. He was one of the really, really big thinkers in developing algorithms. And on his very untimely death, the book, The Fourth Paradigm, was put together that identified the evolution of science and research. The first was experimentation. We would go out and observe. The second paradigm was we began to build theories around those observations. With the rise of computers in the 70s and 80s, we then got into computer simulation. We're in this era of really big data, and it's often referred to as the fourth paradigm. And you'll also see publications out there where it says data, the new statistics. So how do we harness these huge amounts of data, particularly in a system like this, where over the past 20 years, for those of you who are not familiar, many of the decisions were made in federal court. Different groups will come in giving testimony using different sets of data, different scenarios. And the judges there just trying to make decisions based on very eminent scientists who aren't even looking at perhaps the same information or working from the same tools. And of course, more recently, certainly a big problem for us in universities when it comes to grading papers and everything else, the explosion of artificial intelligence. The tools we've been 2 decades, artificial neural, genetic algorithms, but it's now operating at a different level in this era of big data. And so just for fun, we had the meeting DPIC and ChatGPT, as I was a little short of time, and said, just basically, tell me what I should say about big data in about 100 words, because there wasn't a lot of time, and could you write it in the style of David Attenborough? So I sounded as though I knew what I was talking about. So a lot of fun, but basically the 3 points that came out, I won't give you the attempt of David Attenborough, which is actually quite good. But the 3 points were data integration, predictive analytics, and workforce development. Exactly many of the things which we've seen arise. And so if we go back to 2012, when NSF were really making these huge investments, the Exceed program, $730 million just for the first phase, really began to transform what we were doing. And here I took a slide from Alan Blatecki, who was at the time the head of the cyber infrastructure. And he pointed out this, we had 4 centuries of science with constancy, where maybe we were using 10 to the 3, 10 to the 4th data points. And in the past 4 decades, we've now moved to the petascale. And this was a graphic he used to show where NSF were planning, where they were going to make their investments to ensure that we could harness the data. And what's interesting, if you look here from a computer scientist, genomics, climate, and environment. And by 2022, not only were they becoming the biggest areas, the biggest needs, but look at that overlap. As we heard this morning, the role of genetics were just at the very start of the potential of those kind of technologies. So what this has meant is there's an awful lot of expertise there ready for us to rise on. And then I couldn't resist, as he's not here, Wim Kimmerer. I took one of the slides from his presentation back in 2015 when we're talking about the food web analysis. And Steve and Kenny and others can throw up your hands and say, this isn't right. Wim put it out. But here he just gave an idea of the scale if you're going to truly catch the size of, and the challenge of doing the food web. So there's for understanding one species, the number of species you need, and the number of individuals, and the challenge we're facing. And as we heard in the opening day, several speakers just talking about, we're focusing on a few species because that's the responsibility of an agency. And that's where the focus, but we need to understand how species could be interacting to really understand the full picture. And so I'd like to thank Steve for this slide, which really, I think, captures the discussion throughout this workshop today. The ecosystem goals, we need to know where we're going. What are we trying to achieve in this highly manipulated and managed system? And then all of the various drivers, which potentially have an important role. What are the species we're going to focus on? And which are the important interactions? But I think it's important for us to remember we couldn't build the super model to capture all of the food web. But the concepts which we've heard from several of the speakers, you start simple and you begin to build on that over time. And again, from that workshop in 2015, Jay had a really nice slide where he said, why do we do modeling? It encapsulates our current modeling, but we should never stop. As we understand more, you evolve the models, and those models are never completed. They're just abandoned when the next generation of models come along. And so this is a personal, it's not the official, your DSP or council summary, but the meeting on Tuesday was very, very interesting. And this is where very senior people in the state, federal agencies, all of the people that have a role in the Delta had a senior person there. And there were many points made around the modeling. And I just wanted to highlight a few of those. Probably the one that generated most discussion was science communication. And what we heard was that the science needs to be relevant, but the people you're communicating with to what you're presenting. And also many, particularly not just on the council, but they said elected officials, we're the people that go to the legislature. If we can't understand it, we can't promote what you're doing. So you really need to make an effort to make it very clear the science is doing, what the value is, where it's going, and then we can be the champions on your behalf. We also heard about the co-generation of knowledge, and thank you to Zachary for really highlighting that throughout this workshop. There's many, many interests which in the past have simply not been at the table. So how do we pull people together? A third point was made was deepening the bench. Often too much relied on too few through the drought. Some of the models that were trying to turn around and give the best information were just incredibly stretched. We're working 16 hours a day, 7 days a week. I'd now like to step outside the state. I don't want to point fingers, but I'd like to just highlight a couple of problems when the bench is thin. And I'll give you one very talented young modeler who was producing projections of for load reduction across multiple jurisdictions. It had legal ramifications. On top of that projection, suddenly there was climate change and the recognition that all of these entities needed to do a lot more. Most of the low-hanging fruit had been done, so it's going to be really expensive. And then, whoops, there was a mistake in the model. And there needed to be an additional 20% reduction. And without talking about the system, the courage of that young modeler just to come forward, if he hadn't said anything, he'd been through reviews and it hadn't been picked up. But we need to do a better job, and the bigger the community, that kind of thing picked up. To give another example, studying for a potential new reservoir and the capacity. Studies were done, and there was a scenario run, well, let's just make the reservoir infinite. And then the code wasn't changed back, and no one picked it up. And so for many years, that model was being used to project water allocations with a reservoir that you couldn't run dry. So it's what I'm saying is I'm not being critical, but you need to put the infrastructure around the modelers that have this huge responsibility and giving them the resources to have the right checks and balances. We also heard about downstream models. These are models that go beyond just compliance, but when you're putting these sophisticated models together, they can be used for many other things, the HABs predictions, oil spills, etc. And the final point, which we heard from these very senior folks, was please pay attention to governance. And I'm not citing the directors because I couldn't get their permission to give these points, but they said this is just really important. And whether it's the DPIC that set the priorities for the modeling. And I'd like to finish, Steve, I think I've got 5 more minutes. Is that about right? Just the role of tools. And for those of you who don't know Wilson Miner, he's one of the really big thinkers in Silicon Valley. And go to YouTube and hear a couple of his lectures. But his point is that tools change how we interact with each other, how we behave, and therefore how we think. So really thinking through how these models work go way, way beyond just a single projection. And as an example of that, the Snake River Plain water rights adjudication is one of, I think, the most remarkable modeling studies that I've seen. In this, the director of water resources, or he became the director of water resources, Dr. Dave Tuthill, was fed up of seeing neighbors suing one another using the entire family's life resources over an issue that was then decided in the courts. So there was this very sophisticated water rights adjudication was done. And some of the things they did was remarkable. A special court was set up. DWR put out these mobile units, so they weren't relying on people to come to public meetings. They actually went out, they explained the models. The model was developed with, across the board, every interest represented. And to be honest, it isn't a very sophisticated model. But with all the legal challenges in resolving this, there was not one legal challenge against the model. So it just shows when you structure that model in the right way. And here it's much more complicated because we're not talking about one model, but we're talking about integrating multiple models. And coming back to California, Steve asked me to be inspirational. And I think what all of us from outside California recognize that sometimes you don't when you live in a state. The Chinese government does a very detailed analysis of higher education around the world and its role and influence. And in their criteria, California has 6 of the top 25 universities in the world in terms of influence. This results in a workforce and elected officials that reflect the higher education asset. And that results in this consistent commitment to science-informed decision and management. Politics comes and goes, but there's no diminishment in the commitment to science. The mature community I mentioned. And then we have this organization, the Delta Stewardship Council, that oversees the DSP, the Independent Science Board, and DPIC. And because they don't have a dog in the fight in anything, they're just there to be independent to organize the science. It brings transparency, trust, as well as dialogue. And the last thing I'll just mention here very quickly is the Engaged Scholar book. If you haven't heard of that, higher education's really looking. What the Engaged Scholar says, it describes how in research universities, as we advance in our careers, most of us go down a narrower and narrower field, talking to a smaller community using language that fewer and fewer people understand. We pick projects which we can get funded, and these really big issues sometimes get overlooked. And so we're seeing a push by most of the big research universities to be more engaged in the states and the communities. So very briefly, what the Collaboratory is definitely not. The idea that's been voiced in several, it's not a single universal model and Uber database. We know those things don't work. It's not an automated decision system. It's not taking any of the responsibility or the capacity out of agencies. It's value added. And it's not a time sink for the key modelers in an organization. And it was great in Rosemary's presentation this morning, I thought, to see some of the progress the state's made in this area. So what it is, it's an innovation hub. I won't spend a lot of time on this because that needs to be defined. But I'll pick just that middle paragraph. In 19th century Britain, there was a term which for some reason has lost relevance and use, and it was called academic loafing. But what the concept was that we've developed, it doesn't matter if you're an agency or a university, that we've forgotten to have that time to sit, be creative and think big. And so creating the kind of space where you can bring the right people together to think strategically is really important. And then perhaps I'll just skip that in time. So just in summary, I want to say that this is urgent, and it's urgent for two reasons. We've heard from some of the speakers. Climate change, it's unbelievable how fast our systems are changing, and it's accelerating. And I don't know where you're working, but I don't think anyone would have predicted the kind of changes we're seeing in whatever we're looking at. The possibility to accelerate knowledge discovery in a way that protects the agency's sovereignty, it creates efficiency, it creates sharing of some of these tools, and enables rapid response, so that we don't have a state agency director where they talk to their experts in the agency, and then they go to the effort to seek advice from academia and other agencies and make a decision. And then the following Monday, front page of the local paper, scientists say that this was a terrible thing to do, because that will always be there, but it would be nice to have the defense. Well, here's the consensus. It's a minority majority opinion. Here's the data that went into it. If there is this kind of concept, you need the big idea that people will rally around. I think the University System of California with co-equal and the outcomes of this workshop could provide those grand challenges to start. The role, I think, of the science program has been proven. And the final thing that I would say, if this is really going to happen, there needs to be a small, whatever it's called, a pursuit group. Because trying to pull together all of those ideas in a group this size simply doesn't happen. There needs to be just 2 or 3 people coming up with a concept, talking about to philanthropy. And I didn't mention that, but state funding pretty much across the country is going to be down in the next 2 or 3 years. Federal funding is way, way up. And philanthropy is, if you look at the number of billion-dollar gifts related to the environment in the last 12 months, it's remarkable. So why shouldn't there be a concept here that could be funded with that kind of backing. But it needs a small group. This is your number one responsibility. You have the time, you have a timeline to come up with a recommendation. You vet it with the science community and the resources to do that. So with that, Steve, thanks for the opportunity. I'd be happy to take any questions.
Thank you. Just one of the points that Chat GPT gave you, and in your deepening the bench, talking about workforce recruitment and workforce enhancement, and you said we need to do that. Do you have thoughts as to how?
So I think, yeah, first of all, if we just focus on the workforce development, I think in the last few science meetings, people have asked, how many people are thinking about retirement, and it's been staggering. We did it in, I think, was it 2015? So we know we have this need. How do we do it? I think comes from two ways. Your NSF has a program specifically focused on training scientists trained in a certain field to be able to work with big data and big models. And I think looking to that kind of funding, I think building the network, perhaps in a place like DWR, of offering the type of fellowship through the science program or Sea Grant for people to come in and bring their experience, and then it's a great opportunity to road test. But I think it's something that right now we have the data scientists, super well trained, and we have the scientists, but very, very few people have the ability to cross that boundary.
I guess when I asked it, it's a slightly loaded question because I have opinions on this, a lot of opinions. And I think some of the work that is being done in the agencies right now is really aiming towards that. DWR has recently partnered with the Data Lab at UC Davis to put on a lot of trainings, and even just though the caliber of the entry-level environmental scientists has really increased in their knowledge of data science and their ability to work with statistics and code. The other thing that some of these collaborative groups that already exist through IEP and other formats, those are just the ones I know best, are sort of support groups for okay, we're all scientists who need to work with this data now, but we're not trained in it. How can we help each other and lift each other up? In this room, I think the number of people near retirement is maybe the larger percentage of the group, but we've got a lot of really good young, excited people looking to take this on. So just wanted to highlight that, and also starting even before you get to the entry-level positions at a state agency, programs to increase that science communication with students at the grade school level and get people excited. The Interagency Ecological Program has partnered with the school in Rio Vista to get our scientists into their 6th, 7th, and 8th grade to teach them about the food webs and community and science that's going on right around them in the Delta. So I'm glad that you highlighted that need, and just wanted to let everyone know some of the stuff that the community is doing along those lines. Thank you.
That's great. And I think just deepening the state agency university partnerships is incredibly valuable. So
Peter, your very last point there on having a pursuit team to follow up on or to act as, I would say, instigators of how to really pull things together. Could you say a few more words about it in terms of the characteristics of what that pursuit team might look like and what sort of resources they would need and how it might be structured and what a timeline might be? Just give us your vision, if you can, of what that would look like over the coming couple of years.
Sure. And I'll give my personal opinion. There's probably a lot of experience around here, Stan and others, that have really gone after these big, really big competitive federal projects. And I think in those projects, you need the people that represent their particular community. The science program, I would say, has the staff that can help coordinate that. But having the 3 or 4 folks that are trusted and respected by their agency, their interest, I think, is really important. And also it needs to be the type of people that are inspired by the concept. And particularly if there's going to be pursuit of philanthropy funds, tracking the large competitive research programs, there needs to be someone with that kind of expertise. So personally, if it's not my choice, but I think the science program, ensuring there's the diversity on that small group that can reach out and articulate what the concept is key.
That I think is a really good approach. I had a slightly different vision when you were talking about putting together a pursuit team, because it seems to me you not only need what you just described, but you also would need, in a way, the leaders in industry, the really big thinkers, and those who have the connections at appropriate scales, commensurate scales that can bring in the kinds of resources, both financial resources and the resources in terms of people, to really build the capacity for a collaboratory or something like it that operates at a very high level. And those are very special individuals, and ones that are not sometimes they're not easily convinced that they want to join such an effort. And so if you have any thoughts on, how you would put together that team, whether it's a team of 5 or a team of 20 or a team of 1, that would be very valuable, I think, to us and to others to begin to think about this. So,
yeah.
Thank you.
Actually, I know we don't have a lot. If I could just do a very quick response to that. I think you're absolutely right, but that's almost the second step. Because if you don't have that compelling statement and the exciting vision, you're not going to have the Microsofts, the IBMs with any interest. But if it's something that's, say, being vetted by a community like this, that this is really going to make a difference, it's going to move the needle, it could be a model for other systems. Then I think once you have that concept, that's when you can talk to these deep thinkers, the folks that are guiding the strategic directions of those, whether it's industry or the really big philanthropy. But there's a lot of competition for that dollars, and this first small group that I'm talking about has to have the ability to make an exciting and compelling argument.
Oh, I agree. Thank you.
Well, thanks, Peter.
We have time for one more.
Oh, sure, Mike, go ahead.
Yeah, thank you, Peter. That was informative, and I thought it was inspiring. And the collaboratory dialogue here is still a work in progress. And I know you've been working with others on the East Coast on similar ideas. Given that the core objective, the core goal of this effort is synthesis, it's not really modeling. Modeling is a means to an end. Can you share any examples of collaboratories or collaboratory-like institutions that have arisen elsewhere that have enough of a track record that we could look to them for some more insight as to what the secret sauce is that makes them work well?
So Actually, Mike, that's a great question. And with some of the logos that I put on there, I think they're not exactly what we're doing, but I think what NCS has done, what Succinct did, what Data One did in different areas, we've learned an awful lot from that. And I think we're looking at something just a little bit different here than any of those entities, but there's a lot of lessons that can be brought to it, as well as perhaps some logos, as was said in our breakout session, of things why hasn't it worked when these kinds of concepts have been put together. And certainly on the East Coast, we did talk to some of those groups. But I would say there's just a lot of experience around this now that I think could be brought to what's already a pretty highly functioning community.
Yes, I too want to thank Peter for what I also found very inspiring. Just exactly what I hoped you would set us on a track for. We are now in the last part of the workshop, and the good news is we do plan to end early. So all depends on you, of course. The next section is the last breakout, and perhaps in many respects the most important breakout. You only have one question, and that question is, in the Delta, what are the top 3 things that we can do to make progress on food web science or modeling in the Delta? And then, of course, to prioritize these ideas. So I think it's what we're looking for is we'll have a shorter breakout session, and then for each of the groups to come up with their top 3 things. If the assumption is that we all believe that some effort in terms of food web science or food web understanding needs to be done, and if so, what would be the top 3 things we could do to make progress in this field in the Delta? And then we'll come back and have those presentations, and then we'll have a general discussion on those results, and then we'll close out. So I guess we're breaking immediately into those into your 3 groups again. It'll probably only be about 15 minutes is my thinking, but Edmund will wander around to see how well people are doing on the topic. Okay.
So I know we're all looking forward to the report outs from the stimulating discussions we had at our breakout session. So we have 3 report outs from our 3 panels, 3 breakout groups here, and then 1 from the online group. So we'll start off with Christine.
Okay. Is this on? Okay, all right. So we talked about a lot, so I hope my group will forgive me for focusing on the high level key points for all of the different questions. So when we discussed the types of models and tools that would be most effective, kind of the first response to this was, well, it depends. Like, what are you going to use those models and tools for? So there's a little bit of it seemed to me like there was a chicken and an egg situation, right? Like you have to understand what the question is before you start trying to decide what it is that you want to use to answer those particular questions and the types of models and the types of scales that you're going to be needing to look at. And so there was a lot of interest in moving beyond these linear relationships that are almost like knee-jerk reactions of, there's a fish that we're interested in, and so it's linear relationship. And so there was interest in, can we be more comprehensive? And so can we move into the types of models where we are representing trophic levels? There was a lot of interest in doing that. And my understanding from my group, I'm a modeler, so, but my understanding is that there are currently projects in place to do this. And so hopefully we'll be seeing some of that work sooner rather than later. And so we didn't spend as much time on kind of the second question, but we did spend a lot of time talking about an ensemble model approach. There was this idea of maybe a hierarchical approach for how we do modeling the Delta would be more useful. And so this was referenced in some of the talks and conversations today, where it would be more useful to have these simple models where we're not investing as much resource in them, so that if we falsify those hypotheses, we can move on to something else. And so if we have a set of these small scale models that are across the Delta, we can use the understanding from those models to then feed into these larger, more complex models. So the idea of having not one model and maybe making sure that when we talk about food web models, that we're not saying food web model and kind of implying that there is one model that we're trying to build towards, but rather accepting that we might have multiple models doing different things, having multiple different applications, and that's okay. And so the last thing that we talked about, we spent a lot of time talking about barriers, and I'll circle back around to the point that we made earlier, but the main one I think is worth repeating because it actually occurred yesterday with a different set of people, which is that we need a type of strategic plan. We need some kind of shared idea of why it's important to do food web modeling and how food web modeling, whatever approach we decide to use, addresses this strategic plan. And this is important for shaping priorities and making sure that there is staff and funding and resources available to do the monitoring that we need at the scales that we need to answer these large questions. Because that was one of the key barriers that we discussed first, is that many of these models are data hungry. They require a lot of people going out into the field. They require specialized skill sets in being trained in how to code and how to collect this data. And so there's a backlog of information, there's a backlog of synthesis. And so having that strategic plan and kind of making sure that people are aware of why it's important to continue these food web models is important for making sure that we have the resources to address these questions. I think that is pretty much most of the high level stuff that we talked about, but if my group wants to speak up, feel free.
And we can hold off till the next presentation, Lillian.
Okay, so I will echo that our group, essentially for all 4 of the questions, the first answer that was always written down was, well, it depends on your questions, or well, it depends on what you're interested in. So with that caveat, we'll move on to the other things that we discussed that Christine didn't cover. So one thing that I thought was very interesting that came up in the first question was talking about not only, the motivation behind the models, but different types of models. So looking at the difference between strategic models and tactical models, and differentiating and making sure that when you're creating those models or developing those models, you're differentiating between those goals. Because they can be used for very different things. So for example, you're not going to use strategic models to create stock limits. And so making sure that you're differentiating with those models might be really important. There was a thought that maybe there is enough data to, like Christine's group said, to use kind of these groups of models to better accurately represent the heterogeneity of the system. And it was suggested that, several nice models, so the intermediate complexity model types might be appropriate for doing that. Let's see. So also there was talk that in the Delta, a lot of the modeling is done for compliance reasons. And so again, just making sure that the objectives of the modeling is apparent when it's being developed. We also spent some time talking about modeling uncertainty. So actually that's more along the lines. So now I'll move to the second question. What level of complexity do food web models need to have? So, again, we need to look at multiple scales, functionally different ecosystems, and integrate over multiple spaces and times. That kind of gets along to using a suite of models. But let's see. So we also discussed that there are kind of differences in short-term predictions versus equilibrium calculations. So looking at different time horizons and making sure that you're really differentiating between those is going to be really important. And again, these all have to be tied back to your management goals and your key questions. So we did have some ideas about it would be interesting to develop different scenarios that you test regularly. So you could either use the same model and test it at different time points, or you could use different models that you can kind of compare the results to learn about the accuracy of those models through that. And then moving on to the data that are needed to develop the identified models, again, with the caveat that it really depends on what you're going for and looking for. We talked about some of the new tools that were talked about today, like genetics, and also looking at having additional data and updated data for diet composition, as well as estimates of large predator abundances and kind of spatial location data would be really important. Also monitoring fish passage through open areas or kind of restricted areas like junctions. But we also acknowledge that there's a lot of data going on in the Delta that can be utilized, and we all heard about that today. So in this is a section where we had kind of started talking about some of the modeling uncertainty. So really being careful to understand when you're sticking within the capabilities of the model rather than going out to kind of the tails of the model where it may not be as accurate, but also really understanding the uncertainties of all the inputs and what the outputs of the model are. And then for barriers, we kind of ran out of time, but we generally thought that there is capacity in the basin to do some of these things, but really something like a collaboratory may increase capacity to understand all these things. So really having a consensus about the goals, the management questions, and kind of the priorities would be very, very helpful. In addition to just understanding the finances of how to do this and what's feasible.
Thanks, Lillian. So Kyra, do you want to go ahead?
You two ladies are a hard act to follow. It's not the best to go after you two, but I'll do my best. So in terms of the first question, what models and tools might be most effective, there were some sticky notes that said, things like hydrodynamic models and bioenergetic models, but I think kind of the broader discussion for this section was around, again, what kind of question are you asking? And there was definitely a consensus that the management questions that we're currently facing are maybe not, we use the word crisp enough. Maybe we need to focus a little bit more on honing in on those and exactly what we want to manage and what are our goals, and maybe figuring that out first before we get to that next step of, okay, how do we model something like that? So there was kind of a broader conversation about, the management questions that we're asking, and what we want to get out of that before we even get into the modeling bit. Let's see. And then in terms of the level of complexity that a Delta food web model might need, there was various sticky notes that had, spatial scales in terms of habitat type or region, temporal scales being either tidal or monthly or annual. So there was variation in that. But again, we had kind of a larger, more broad conversation about, you need both the simple models and the complex models. It's not necessarily one or the other. And there's different ways you can go about that in terms of starting on the simple side of the model and building up to the complex, or doing the reverse where you start with the complex model. And then depending on what your research question is, then kind of shaving off parts of the complex model until you get to the simple. So there's kind of various ways to get there, whether you're starting simple or starting complex. But there's definitely a clear need for both. So that was kind of the theme for that question. And then what data are needed? There was a lot of different things listed. There's, diet data, temperature, turbidity, gear, efficacy, biomass. There's a whole bunch of different things listed. But a couple key things that were talked about were that it's unclear how much diet data are out there. There's not a centralized database for that. That's maybe something that could be put together or standardized and would maybe relieve some of that lack of data we're maybe seeing in that region. And that we have a lot of prey data, but we don't have a lot of predator data. So kind of those are the key places that we might be missing. And let's see. So our barriers question, that was the section that filled up very quickly with sticky notes. It was very easy to get some answers on that. We didn't necessarily discuss it as much, but there was a lot of different things listed. And so some of those were resources and imagination. I kind of liked that one, maybe widening our imagination on what's possible. Another one I liked said, need to convince people that this is not scary. So I think that was kind of a nice way to put it. Let's see. One said time, that's a barrier. It's running out on us. Let's see, conceptual models of non-listed species, inability to predict management actions, contaminants, data diversions, reluctance to change, lack of information on food quality. So there was a whole laundry list of barriers. So that one was pretty easy to get information on. So that's kind of just a very quick overview of that. So I'll stop there and we can go to our virtual group.
Thank you very much, Kyra. So we'll go to the virtual group now. And I think, Coco, if you just start talking, you'll show up. Go ahead. Go ahead.
Hi, can you hear me?
Yes.
Good. My name is Shoko Shinbrat. I work for the Delta Stewardship Council and happy to be joining you virtually. I am happy to see that a lot of the things that we discussed have actually already been captured. But we started our discussion in kind of a narrative format, thinking about, what is the appropriate level of complexity. One of the speakers advocated for, as simple as possible. And often, we talked about how systems are so heterogeneous, where complex models are good for, these high spatial and temporal resolutions, but we need to be careful if we don't have the data to build out the model. And similarly, I think this might have come up in every group. We need to make sure that the tools and data that we're collecting really align with what our questions are. So what are the questions that we're really trying to ask or to answer? And then we started talking about making sure that whatever model is built is actually truly representing the system. So making sure that there's validation and calibration of the models. And it's funny, sometimes in these discussions, you end up getting more questions than you do answers. So many of my kind of key points here are actually framed as questions. And in, thinking about uncertainty, particularly, to what extent can these models be used for anticipating major changes to the system? This kind of dovetails with other efforts that the ISB is working on in decision-making under deep uncertainty. And kind of similarly, how can they capture and, reflect tipping points? In addition, we noted that, and I think other speakers have also noted that we're managing for a lot of listed species, but we might lose opportunities to manage for non-listed species. And then we took the opportunity to come up with some questions for the speakers, since we haven't had as much opportunity to chime in with questions. So some of the questions that came up were, to what degree have these models been useful in ways that go beyond what is expert knowledge? What are the management questions that need to be answered? This came up earlier. And to what extent can they be used for anticipating major changes? And then were they the best tool for the job? How does the design for the model matter, and maybe examples that have proven particularly useful. And then we closed out the discussion with major barriers, and just getting back to this question that has come up again and again of are we asking the right questions? All these models are designed to support decision making, but perhaps the questions are not necessarily what is needed. So this is a real barrier that we saw could be resolved through sustained engagement and sustained communication. And that's That's all I have.
Thanks very much. And right now, the plan is to have a coffee break in 2 minutes. So, but we want to open it up for 1 or 2 questions before we do that, or some crosstalk across the different groups. Does anybody have a comment or a question they want to pose? I don't see any. Okay. Go ahead, Rosemary.
I just wanted to respond briefly to the question on how whether models give you more information than just expert knowledge would. And I think the value a lot of managers find in having a model is you can quantify it. If you're writing an environmental impact report or an incidental take permit saying that increasing the amount of pumping will be bad for salmon because Ted said so, doesn't hold a lot of weight, but saying we have this peer-reviewed, quantitative, complicated, multilinear Bayesian model that says increasing pumping will decrease salmon survival by 15%, that's something that holds a lot more weight with decision makers. Even if that 50% has huge error bars on it that often gets lost somewhere along the way, still having something you can quantify and then compare, okay, is increasing pumping gonna decrease salmon more, or is raising the water temperature or increasing harvest, like which of these has the potential to be worse? So, yeah.
Thanks for that comment. Steve, do you have a comment?
Yeah, just a little bit in response to that. I think one of the things models can do is really help you to see sort of counterintuitive results. And I think that we're commenting about the expert opinion. Some experts can be wrong, and so can models. But I think that just looking at the example that we did on the Gulf of Mexico, where Kim and I were using a completely different approach, and I think common wisdom was when you cut nutrients by 40% and have all these improvements, you're going to see major changes in what's going on with the fish. And we both were coming up with 3, 4, 5%. So much so that I know I looked at my data many, many times to see where did I do wrong, and she came up with the same numbers independently using a different approach. So that's the kind of things that I think models can give you, is some of those bounds, some unanticipated results. You might have one species over here that is doing something that you never expected because of a secondary impact or something like that. So that's another value of these sort of approaches.
Stan?
This morning, Ryan made the comment that you learn so much when your models fail. And we rarely get up in these meetings and present the failure of our models. Has there been a retrospective of the models that have been used on the Delta and the failures and what was learned from those failures?
Yeah, this is Kim the Mustard. I can provide one example where, an ecospace model receives input from other models, and it's a food web model. So nutrients inputs has usually a strong effect. If you have more primary production, you have more secondary production. And I think it was useful already, regardless of what my output was for decision making, to noticing in my model that there was much more nutrients in the system than that was possible just because of the secondary production I was seeing, and going back to the models where that came from, where I was like, can I look at that data, right? It's just couples and then looking at, well, there's no way there's that much nitrogen in the system if we make these changes. And it ended up changing that model to see like, where is the mistake? And then we can run it again. So I think sometimes by coupling these models, you can indeed, by crazy outputs, figure out like something along the way isn't going very well. And you can go back and figure out what it is and try and change it. So yeah, I think definitely some instances where models failed have been useful in the process that way.
One more comment or question over here.
Just wanted to add about Vamsi, Sridhar, and Tetra Tech. I just wanted to add about the value of these models. So even when you have intuition or expert knowledge that something will happen, quantifying that will actually lead to proper management action rather than some sort of strategic idea about what you need to do. So I'll give you an example. So with the EPTM model, we were able to look at, it's not published yet, but we were able to look at different scenarios of pumping levels in the South Delta and saw that, at certain pumping levels, the entrainment envelope of fish is at, a certain point in the Delta. At a different pumping level, it's at a higher point in the Delta. With a calibrated and validated model, when you have trust in what the model is doing, you can then take that to management action and say, maybe we need to put a barrier here so that in this particular place, so that fish do not get entrained into the pumps. Whereas with just expert opinion that if you increase the flow in the pumps, fish are going to get entrained, you really cannot say where you need to put that barrier. So that level of management precision, if the model is calibrated and validated, is definitely very beneficial to have.
Thanks for that comment. And this will be the last comment or question.
Yeah, Stan, thank you for your question about model failures. I don't know of like a synthesis of them, but it did get me thinking, and it seems like I can think of an old example that the entirety of striped bass population dynamics could be summed up in a linear regression. That was a model of the Bay Delta system at one time that did not hold up very well. And now I think maybe the newer one that I'm still waiting to hear about is there was a hypothesis for years and some very complicated models developed around the idea that the overbite clam had not wiped out all the chlorophyll production, but that a wastewater treatment plant in Sacramento was dumping out too much ammonium, and that was doing it. And so that led to treatment plant upgrades. To my knowledge, it hasn't led to phytoplankton blooms, but maybe there's a bunch of top secret information I haven't heard yet. So that might become one of the more famous examples of a model fail in the Bay Delta.
Well, as an environmental engineer, I'm going to say that whenever you upgrade a wastewater treatment plant, it's a good thing. So
not arguing that part.
So on that note, let's take our break.
Well, welcome back, everyone.
Billy, are we ready to go?
Okay, if everybody could have a seat, we'll get started. I don't know about the rest of you, but we decided we needed one more day on this workshop. So with alcohol, right. We're going to start with the online people. They're well ahead of us. So Shoko, if you want to present your meeting result.
Sure. First, can everyone hear me? Can everyone hear me?
Yes, yes.
Okay. So I'll do my best to capture our discussion, which you can see at the top that if you give some academics some constraints plus myself of 3 things, that it turns into 3 plus a few more. So we had some really fruitful discussions around kind of 3 broad topics with some details within. So So our first, so what are the top 3 things that we can do to make progress on food web science and modeling? Well, one of the most important things to do is engage with a diverse array of interests to identify what questions the Delta could benefit from in terms of added and enhanced food web modeling. And of course, that's always based on what data is even available. And these can be existing questions and new engagement to generate new questions. So that was our first one. Our second question, or thing that we can do to make progress on food web science is evaluate the types of modeling that could actually support those questions. And then to do that, comparing the modeling goals and again, what data is available, and evaluate feasibility of actually filling gaps in the data. And then finally, kind of reflecting on what we've heard today, and then other systems. Our third thing is to compare lessons learned with other estuarine systems, and considering some of the contextual similarities and differences, whether these be regulatory or species, so biophysical or social. And then thinking about to what extent were tools actually used in the decision making, and then whether there are generalizable and transferable conclusions from these well-studied areas like the Great Lakes or Chesapeake Bay to more understudied areas and species. And that's all from our group.
Okay, Christine.
Sure. So I think this is going to echo some of what our group talked about in the earlier breakout group, but for us, we really reached consensus really early that one of the major things that we can do to move progress along is to have a strategic plan. Thank you. Is to have a strategic plan that makes the case for why having food web models available really accomplishes our goals. And it was important to define and to use that strategic plan to define what we want the food web models to do. So that was high level. Within 30 seconds, we reached consensus on that. And then our second kind of point we wanted to make was that there are some things that we can do right now. We can start with the data that we have to build an empirical model, and then we can apply some of the approaches that we've heard about, such as scenario analyses or sensitivity analyses, to try to identify what are the additional steps that we might need to look into. And then the last point that we had is that predatory fishes have popped up again and again throughout the day. And so that's probably like not an easy identified thing to do, but it is probably one of the things that we need to really increase our monitoring on if we're going to fill an important data gap in the current data that we have.
Okay, Kyra.
So yeah, I think some of the things that we came up with are definitely shared by some of the other groups. I think I'll maybe start with my second sticky note, which is we basically decided, like Steve said, we need another day of the workshop. So we basically we're talking about the need to build a conceptual model or models, which maybe could be done via another day of workshop, which could be a future event. And that in turn would help us kind of clarify some of these management objective questions and sort of hone in on what exactly we're wanting to accomplish and how are we wanting to do that. So that's kind of one key part of what we somewhat are forming into one thing. And then another part, which we've heard from other groups, is the data collection and sharing aspect. So identifying the data gaps and then figuring out how to share the data that we do have and maybe some sort of collective space. So that's been talked about a little bit before already. And then sort of a general increase of resources in several forms, whether that's funding a dedicated modeling team or increasing computational and human resources and then synthesis efforts. So kind of mushing that all into one thing, which is to say just increasing resources around this topic is another important thing. And there were a lot of other really interesting things that were brought up that do not succinctly fit into these three categories. So I'll just leave it at that and hand it off to Lillian.
Okay, so the three priorities that our group came up with were to develop a strategic prospectus and implementation plan that covers a discrete time horizon and accounts for a diversity of priorities, relies on a proof of concept, interdisciplinary approach, clear goals, and implementation of co-management opportunities. So it's a mouthful, but there's a lot of good stuff in there. Second, to develop a collaboratory that serves as a place and a process that can facilitate broad community engagement, learning, experimentation, and observation. And then finally, to have enough support or resources to do this and implement the plan.
Well, thanks for those reports. Now it's time for having an open discussion. Anyone who has any questions to the reporters, anyone has any ideas that were missed, or anyone want to discuss some of these or other topics before we close out the workshop? Yes.
Vamsi Sridharan, Tetra Tech. I do want to kind of throw out there a lot of the groups that were discussed were state agencies, universities, research centers, Indian tribes, other stakeholders. Does this group see a role for the private sector, for consultants in this whole process? And if so, what would that potentially look like?
I think Rosemary has a response.
Just off the cuff, yes, definitely. Anyone who is good at modeling things will probably could potentially have a role. I think just based on the way I would conceptualize moving forward would be, starting with clear management driven questions. And that's going to come from the people who are doing the ecosystem management. Then, that plan and initial hashing out would probably come from more agency, tribes, stakeholders, community. I would sort of assume that, and that process could be facilitated by people from the private sector with contracts. We've had a lot of really good success with having external facilitators help those kinds of conversations. But then once the actual modeling starts, I know DWR frequently uses private sector consultants who are experts at modeling, the work that RMA or Anchor QEA, Tetra Tech, other groups, certainly. If there is, once we have a clear need, then private sector modelers are definitely part of that bench, and talking about deepening the bench. And sometimes agencies are thrown into a drought with 10 million other things to do. And so being able to contract with outside people can be very helpful.
Before Diane talks, maybe this will help a little bit in terms of summarizing what some of our next steps might be in this overall process. Is that we certainly intend to write up the results of this workshop. Whether that will be a separate report or a report that becomes part of our sort of broader science board review on this topic, we're not certain yet because the workshop is still running. And then, but in that process, we have ample opportunity for public comment. We may consider other options that we haven't yet discussed, like having further community engagement opportunities to get some feedback from things we might produce. Our reports then, when they're finalized, formally go to the Delta Stewardship Council as a recommendation. and as often in the form of recommendations and summary. And then, of course, they're widely distributed and what agencies do with those recommendations. And that is something we hope they entertain and take steps forward to make those, not just agencies, but anybody who reads the report to take those recommendations and ideas and move them forward. And so that's open to anyone. Diane?
What I was going to point out is to follow on Peter Goodwin's point about communication and science communication is that involves not only scientists explaining and conveying ideas, but scientists listening to a broad range of individuals and coming from different sectors, the indigenous communities, and setting up those dialogues is an important part of, or will be an important part of developing strategic plans for modeling, because there's members of the community who may not be here in this room who are very interested in the outcome in terms of not just the Delta smelt and salmon, but all the different sport fisheries and such.
Okay, anyone else have any discussion or comment or questions? Peter?
Yeah, Peter Goodwin again. And I just add one thing. We spoke briefly about workforce, and I think it's really important that we consider the diversification of our workforce too, that if there aren't scientists at the table that understand the communities that so many of these environmental impacts are affecting, not only are we losing a huge part of the talent pool, but of course, it's very, very difficult to communicate. So I would certainly hope that with some of the things, Rosemary, you were talking about internships and things like that, working with the universities, I think showing the career opportunities and the impact that could be had would create a much more functional and productive workforce a decade from now.
Up front.
Hi, Dan Ellis from IP. I was bringing this up to the group, and it's pretty much just tailing off the establishing clear management objectives. And I wonder if it might be time to engage the public in an honest discussion of what the resources can provide and what they can't moving into the future, because I don't foresee people being happy right now. And I wonder if maybe it's just time to sort of like set a new vision for the Delta that has like some actual chance of us achieving those goals.
Any other thoughts? Yes.
Thank you. I just had some closing remarks I wanted to make. I actually wanted to read a brief passage from this book. I didn't have time in my presentation, so forgive me for saving this for the end of the day here, but this book is called In the Absence of the Sacred, The Failure of Technology and the Survival of the Indian Nations. The author's Jerry Mander, and I felt that this passage was highly appropriate, but I'm going to ask for your forgiveness in advance, because what you're about to hear is probably anathema to the high-tech modeling approach we've been talking about this whole time. And I'm not asking you to become a neo-Luddite. I am just asking you to walk in both worlds with me and diverge from the black and white thinking that we often fall victim to. So it begins, it seems quite obvious, almost self-evident, that native cultures that have lived successfully in one place for millennia have been abiding by successful economic practices, including wildlife and resource conservation. But if we listen to our Western scientists and governments, we would think that native societies can barely manage another day without computers, quotas, satellite mapping, and maximum sustainable yield analysis. How, I wonder, do scientists rationalize how natives have survived for thousands of years? Instinct? The assumption that our modern system of wildlife and resource management is more efficient, despite the fact that we manage without any understanding of the environment or the way that people have managed prior to our arrival, is not only hubristic, but racist. Now, this is a quote from Milton M.R. Freeman, who was speaking at a conference in the 80s. And this book is quite outdated. I will say it's from the early 90s, but I find some of these passages are very prophetic. He said, an explicit faith in the correctness of the scientific method is so integral a part of the professional formulation of wildlife biologists that the limits of that particular system of belief are only learned often much later in life as a result of experience gained in the non-professional world. And there's a very interesting example here that I think really elucidates the knowledge traditional tribal communities hold that can be missed by scientific analysis. So one example concerned caribou hunting on the Ellesmere Islands of Arctic Canada. Canadian wildlife managers told the Inuit that they should hunt only large and/or male caribou and only a few animals from each herd. The Inuit argued that this practice contradicted their traditional relationship with the animals and would destroy the caribou herds, but their pleas were ignored. The result was as the Inuit predicted. Though their new limit was only 26 kills per year, far less than an Inuit had hunted before, the formerly abundant population dropped sharply. Why? According to Freeman, the Inuit hold that each small group of Peary caribou is a social group and there's a good reason for those particular animals being together. Inuit hunters point out that given the marginality of the environment for herbivores, older, larger animals are important to the survival of the group. These older animals have experience and they have the physical strength enabling them to dig through the snow for food. Old animals are also more passive relative to the more nervous younger animals or pregnant females, and this behavioral trait has a calming effect on the younger animals in the group. And then this last piece really talks about the dichotomy between qualitative and quantitative methodology. For our present purposes, it is sufficient to observe that, as in the case of the Peary caribou example, behavioral knowledge of the species was the critical point of the Inuit position, contrasted with an inexact quantitative perspective proposed by the Game Management Service. In reality, both native systems and Western science rest on the same foundation, namely empirical evidence. Both systems place value on the systematic accumulation of detailed observations and the abstraction of norms from disparate data sets. At this point, however, the two systems begin to diverge. The native system assesses deviation from the norm in a qualitative sense. For example, animals become fewer or fatter or more excited. There are fewer calves in the herd, more injured bulls, more barren cows, etc. The sum total of the community's empirically based knowledge is awesome in breadth and detail and often stands in marked contrast to the attenuated data available from scientific studies of these same populations. So thank you for bearing with me as I read that. I just had a couple closing comments that were really inspired by Peter's presentation. I wanted to emphasize to all of the folks in the room the importance of tribal access and co-management as we move forward. Our interest in collaboration. We have so many hypotheses that we would love to test with the research community. We've just barely scratched the surface of these investigations. And really, it's not just a matter of equity. This is the solution. This is the pathway for navigating the ecological and biodiversity crises that we see today. I feel indigenous knowledge and traditional stewardship practices are really the key to finding a sustainable future. And the way I see it is we have a lot of common goals in this room. Science is one leg, traditional ecological knowledge is the other leg. If we walk on two legs, we'll get to our common destination a lot faster. And some folks have mentioned, the focus on listed species. I want to emphasize that tribal collaboration is not just about human equity, but ecological equity. And we care for all our relations. It's not political, although things can be made political when we talk about our relations. It's not based on listed species. It's based on thousands of years of relationships, and those things are much harder to politicize. Finally, I really liked Peter's quote about drowning in data. I think we all experience that, but I would like to make the case that tribes have had the depth of time to process that data and arrive at wisdom, and we can help to provide that wisdom. And then lastly, there was that quote you mentioned about the tools shaping interactions, and it actually reminded me of a quote by Marshall McLuhan, who's a preeminent media scholar of the 20th century, and he was talking about the nascent introduction of television, and he said, "The medium is the message." My point here is that when we embed ourselves in technology, we're actually changing our perception of our felt environment for better or for worse. And so we need to interact with the world beyond the models. And tribes can help bring this non-reductionistic view because we really see everything as being connected. And that can help ensure that we're asking the right research questions. And lastly, sorry, Peter, climate change was predicted by Native people, just nobody listened. So thank you, everyone. It was really an honor to be here, and I'm so grateful to share this space with all of you.
Thanks for those comments, Zachary. Any other comments, questions? Okay, before I turn it over to Engo to officially close our meeting, I want to thank a few people. I certainly want to thank Lillian and Christine and Kyra and Edmund and Lauren Hastings. Those are the folks that really made this meeting happen from the very beginning of this process to through all of the various community engagement processes we went through and putting everything together, including the briefing paper. I also want to thank Bob and Diane in particular as my science board colleagues who helped coordinate all of this. But I guess I also want to make sure I thank all of you for hanging in there, but all the speakers and all of the discussions. I do think you followed our instructions to be mostly optimistic, and except for that one, barrier diagram. We had so many Post-its on that. But I think that I want to thank everyone. I think you provided a tremendous amount of information. I think we really think this has been a very successful and an overload of really good ideas and information. I think we'll have a challenge to put it all together in a reasonable and valuable format. So, but I do want to thank everyone. And with that, I'll turn it over to Enga to close us out.
Thank you, Steve. You took all the thank yous away from me. What remains to be done is thank you for being very instrumental in putting this workshop together. I thought it was super interesting, and I hope we will find a path forward to coming up with a great food web model in the future. I need to ask if there are any more comments that are not on the agenda.
There are none.
In that case, all I have to do is adjourn the meeting for today and wish you all a good trip home, and or maybe another night stay in Sacramento. And thank you for being here.